Welcome to my keynote on in vitro neural stimulations. And I thank you for the organizers for asking me to do it. And I guess when they asked me to do it, they really wanted to maybe bring something different for this brain stimulation conference. So my topic today is stimulation assessing emerging in vitro neural models. So just to give a little bit background, what my group is doing at Tampere University. So my group is kind of having three different main cell targets. One is epithelium, one is neurons and astrocytes, that is today's topic, and third one is cardiomyocytes. And what we do is three different things: in vitro methods for electrophysiology especially, and 3D imaging, especially tomographic imaging on mesoscopic or cellular systems, and computational modeling and analysis, all these. And of course, today's topic is neurons and astrocytes. And I'd like to also introduce two projects that I'm involved. One is Finnish Academy Center of Excellence on body-on-chip research. And it kind of spans from epithelia, especially neurons, but also cardiomyocytes, but other tissue types. And another one is HERMES, that is a hybrid enhanced precision medicine system, and that is a European future emerging technology project. I introduce this here because you will see some slides with these logos, so you know what these mean. Okay, so let's look a little bit what I'm going to talk you. First, we'll look a little bit about in vitro stimulation neural models. So why we need to have these models. And then we look a little bit about the engineering behind these, because actually there's quite a lot of different engineering. And then we look the biology side. So what kind of models we can have in vitro, and a little bit what are the kind of challenges and what kind of stimulus system we can have there. A couple of examples. And then future directions. So another one is really looking how we can kind of improve the biomimicking complex systems. Another one is really looking on this in vitro in vivo translation. Okay, so what is in vitro neurotechnology? Is it a brain on test tube? Could you call it brain on a dish? And actually, it's more or less like this. In the brain stimulation community, and most of you are, what components you have? Of course, you have the brain. You stimulate the brain with, anyway, many times with electric and magnetic fields. Then, of course, you have the recording system. You record the responses. Many times it's EEG or MEG, among other things. And then you have the signals. And in principle, in vitro system have the same. We have the recording system that is called here microelectroarrays. And this is actually something I would like to concentrate more on this talk. Then we have the brain, and that is a neural system that lives on top of these electrodes. And then we have the signals. So very much the same components. And of course, this is about modeling. So why we do these in vitro models is we would like to figure out what the neurons are doing. And of course, it's a model of the real neural systems, a way or another. And of course, one key question is that how good are these models? So if you think in general, how we model neural systems, of course, we can have the human brain, but of course, it's somehow limited what we can do with the human brain. Normally, we are quite limited to the outer surface or scalp. In mouse and other animal models, of course, we can go in and do some more invasive studies as well. But many times we would like to go even closer to neurons. And that's why it's sometimes much better to go on in vitro systems. So culturing the neurons in vitro. Of course, here you can have different kind of scales. A lot of studies have done on kind of single cells, couple of cells, where we look the like patch clamp electrodes, the kind of ion channels, or then here it's more like network level. And I think this is kind of interesting level because it's the kind of more relation to the brain, because here it's a network that has also very many similar properties of the brain, even it's, as said, just a model. Okay, why we do it as well is kind of translation to humans, because many of the work done, closer look what neurons do is done in animal models. If you would like to see, for example, drug effects or some other effects of the neural system behavior, you would like to go to more like humans. And in vitro offers this way, because we can actually have the in vitro models based on human cells. I'll talk that a little bit later. So what are targets of electrical stimulation in vitro? There's actually quite a lot of different targets. Starting from cell differentiation, we can think about it if we can boost electric machinery on kind of early phase neurons or stem cells, those could maybe differentiate better the neurons. That has been done. Also, we could think that we could have a better neural growth. For example, we could try to grow axons that could be helpful in certain diseases as well, and test it in vitro. Sometimes it's also nice to actually look what are the adverse effects of stimulus, because there are many. Of course, we can look the stimulus to look the activation, information propagation network. We can challenge the network communication. These are as well the same things done in vivo, like human brain stimulation wells. And of course, we could try to do also network modulation and look like learning events in networks. Then, of course, if we can, of course, learn a lot of things in vitro that we can hopefully translate to in vivo, or build this future bio-ICT hybrids where we could kind of, let's say, have electronics and neurons living together, whether it's outside the human body or even implanted. I have a couple of examples of most of these areas. So what is the technology behind these in vitro systems? First of all, as I showed before, these are the components. We have the same ones that are in vivo: the cells, the brain, the measurement system, and the signals. But of course, there are some similarities and differences. And so those are the same things. Electronics, electrodes to record. We have a brain or the neurons. We have the signals we would like to analyze. And then also in vitro, we have structural and functional measurements. Similarly, as in vivo, we have MRI, functional MRI. Here we have imaging methods, microscopy methods, for example, to stain certain neural structures, or then functional imaging like calcium imaging. And of course, then we can analyze these systems a very similar way as is done in EEG, or also looking at the connectivity on the brain on MRI. But of course, there are differences. One major one is that, of course, if you do human or animal studies, you have your culturing system. You have your animal or your patient that is actually taking care of the brain. But in vitro, of course, you have to build your culturing system and environment. One thing is that in vitro, we don't have any other means to stimulate the system. In vivo, of course, you can use different sounds, all kinds of senses to alter the brain functions. But here, there are none of that. Then we have only part of the brain or part of the neural system. So there's no higher kind of cognition available. And similarly, there's no response. The functional responses are not as in vivo, not kind of, let's say, breathing or heartbeat change, nothing like that. Or there's no locomotion or something else. So all the analysis is based on signals and, of course, the images. And this is just a basic tool that many uses in vitro. That is the microelectroarray. So these are maybe half a dozen manufacturers around the world to make these systems. They are more or less the same, the basic form. So we have this kind of quite sizable dish where you can culture cells, and then we have bottom there electrodes. Like here, the electrode distance is 200 microns. That is quite typical. It can vary quite a lot. And also the electrode distance here is 30 micron, and it can range from a couple of microns to, let's say, 100 micron. So they're pretty small. So they are microelectroarrays. And of course, one important component I'm not going to talk today very much is the electrode itself. Like here is titanium nitride surface that makes this kind of large surface area, almost like a tree structure. But that is very important if you would like to stimulate your cells to have very good current injection capability. And of course, there you have your network growing and you record them. And if you would like to stimulate these networks, normally it's very similar kind of pulses that you deliver, for example, DPS. It's biphasic pulses many times, and those are like average kind of signals that we can use, couple of milliseconds or parts, microsecond pulses, half a volts, 1 volt pulses, and then you get the responses. Not all MEAs are similar. This is another type that's kind of interesting. It's like building growing your cells on top of CMOS circuit. So this is actually CMOS circuit, and on the right, the small dots or round ones are electrodes. They're actually a gate of a FAT transistor that you can grow your cells on top of those, and they record the charge transfer from your cell surface. And there's also quite a number of stimulus sites. So these give quite a lot of different kind of application areas as well. Much more detailed picture, but a lot of work has been done in the basic MEA. And if you think about the microelectroarray signals, it's quite different than EEG, because now we are really recording from the neurons, but it's still field potential. So what you get here is a signal that on the right, and you can see this looking like a noise, and then there are a couple of spikes emerging, and those are the neural signals from the really adjacent electrodes. Normally, we can record something like signals around 3 times the area of the electrodes. There are no volume conductions as same way as in brain or EEG. So it's much smaller electrode area. And then if you think about this signal, the spikes we actually measure, and then or we identify them normally very simple way by having a kind of cutoff voltage thresholding. And sometimes the spikes form this kind of spike trains, or even very dense spike trains, and that are called bursts. And many times the analysis of these signals are based on statistics, how much there are spikes and bursts, and what is the transition between spike and bursts. And this is how they look when you identify the spikes and mark the spikes every time on all channels. There are 60 channels, and you can see these dense groups where the spikes and bursts are, and you can see that they are in every channel time to time. That means that the whole network has synchronous spikes or bursts. So we analyze spikes and bursts. That is the basic things. That the statistics of those. You can look more carefully. You can actually also look the burst analysis and sort the spikes. Like here, when you look more carefully in one spike, they actually differ. Then you can classify your spikes to different forms, and then look how many of those spikes are different forms, and how your stimulus affect them, how the drug, for example, affect these spikes, and how much they are in maybe spikes, and how much different kind of groups are in bursts. Of course, you can also look the spatial distribution of spikes or bursts in your network. And of course, one same kind of thing that you do on the brain network, we can also analyze the connectivity inside these networks. This is one example of those methods that we use based on spectral entropy. But there are many methods for this, similarly as in EEG. And it has exactly the same purpose, to look the connectivity and look the kind of how it's developed over time, for example, here. In vitro is kind of nice because we can do a lot of things. One is that we can, like here, manipulate the cells. We can inject materials, drugs, or genes to the cells if we like. We can, of course, measure them. We can culture the neurons on top of these electrodes. We can have mechanical stimulation. We can have heat distribution. We can bring gases on this system, or we can have fluid shear, for example. This, by the way, slide of my colleague Pasi Kallio, who is mostly doing these in vitro systems for microfluidics. And of course, we can then measure all kinds of things like patch clamping, microarrays. We can even look the migration of cells, or like here, cardiomyocyte speeding. So the image-based analysis, or we can even have kind of environmental sensors. So in vitro system is kind of nice because we can really look how different kind of methods, how to model really biological system and look the environment, have different diseases in vitro on this, maybe build trauma, have different drugs, and maybe also some phenotypes, and really look the mechanism of things much more carefully than maybe in vivo systems. Okay, so what is the kind of some of the main challenges? Of course, there are a lot of kind of engineering challenges to do it, but partially engineering challenges are linked to the biology. And of course, the main question is how we can make these in vitro systems better, how they can mimic better the brain structures. And of course, there's quite a lot of ways to do it. I'll look some of them. One is really to go from 2D to 3D. But of course, another direction is that how to make these structures more defined and repetitive. If you think you're culturing cells on vivo, they actually look very different every time you culture them. There are ways to do it. So this is just a kind of little introduction on what kind of models we have in vitro, because we can have different kind of models. One is ex vivo tissues, another one is culture of dissociated neuron cells, and then really kind of strong emerging area, stem cell differentiated neurons that has been around like last 10 years. This is just an example of work that really started the in vitro system, and that is the ex vivo brain slices. Like here is a rat hippocampal slice, and because there's an injury in this hippocampus, it's also prone to epilepsy. So you can see at the end on the recording how the spiking turns to the more like epileptic bursts kind of activity. And these slice recordings have been a lot of also stimulus recordings where the connectivity of the slices of brain, parts of brain has been studied. Also, a lot of work has been done in the retina because this is an excellent tool for looking at the retinal circuitry. Okay, and then very common is animal dissociated neural cell cultures. What it means is that you have really brain cells from living animals, and then you really kind of redistribute them on the surface of the mare. And for example, this comes out of that. So they regroup, grow these kind of mini areas and connect connections. These are pretty easy because they're pretty fast. A couple of days and you can have this kind of nice culture and start getting signals. And of course, one interesting what we talked already is the human cell cultures. To my knowledge, this is one of the really first papers to show that we can grow human stem cell based neurons on MEA, and we can get nice signals, and they generate also network. And this just shows as well that when you start from stem cells or precursor cells, it's very slow. They start to make connection, a couple of weeks takes, and they have a couple of spikes, and it takes many weeks to get this kind of level of integrity that you can get bursts like in slices. But of course, what really changed the whole thing, these were embryonic stem cells, was the emerging of pluripotent stem cells, and these really changed the world, because now some of the ethical issues were cleared, but also the idea is that we can have cells from any of us. So they're really patient cells that can have diseased cells, because if the patient have some diseased, or then of course we can think about they are excellent for personalized medicine. I think many of you know this, so I don't take more time on these, but these are really available quite widely. You can buy these cells from the shop as well. Okay, but let's look a little bit about this. So these are dissociated rat neurons, and you can see a nice network, some connectivity, not the signals here, but of course the signal as well. But what is kind of bad here, that whether you have human cells or rat cells, every time you plate the cells, they form different networks. So they don't have rules actually how to make the networks. They don't make brain-like structures. And that's why people have tried to really make systems that better mimic the brain, or really look how to make these more kind of defined. And one is really to build this kind of environment where you have kind of network structure emerges. Like you have something like material wells that force the cells to form patterns. That has been used a lot, or some other patterning methods on the surface that guides the neurons to go certain directions. Don't go very much in detail here, but I welcome you to read, for example, this paper that is from Janos Vörö's group. That they made the nice patterning, and also were able to make this kind of system where the neurons are making directional connections, not just connections on a certain kind of way, but also directional connectivity. So of course this increased repeatability and control, but it doesn't necessarily bring kind of more biomimetic systems. This actually one attempt from Tampere, my colleagues Susanna Narkilahti and Pasi Kallio, how they made this kind of microfluidic system. On the left, you can see that there are two chambers, the blue ones, there are neurons growing. And then on the middle, the neural cultures actually make connection, they have actions. And there's a third chamber where they grow oligodendrocytes. And oligodendrocytes are cells that make then myelinization. And of course, this is much more like brain structure, because you have like two brain areas, and then they are connected by myelinated cells, just like the white matter tracks in brain. So this is the step forward for that direction. And of course here the stimulation was made easy. You can stimulate another side, and then you can follow how when the pulses reach another side and go through the network or the tunnels with oligodendrocytes, and you can follow the myelinization by speeding up the travel of the pulses. Of course, another way to going is really looking at 3D cultures, because brain, after all, is not growing on glass, not even inside glass, but still it's 3D, and neurons and astrocytes, other cells, really would like to be on 3D and not in 2D. This actually old slide from, we had this kind of 3D neuron EU Future Emerging Technology project many years ago, where we actually growed, this is again from Susanna Narkilahti lab, it's kind of neural cultures that start to emerge on 3D hydrocells. And that is the famous, most kind of used material for scaffolding on 3D neurocultures. What is now emerging actually is this kind of organoid structures, and these are structures that kind of neurons, with treated properly, they start to make a more like brain-like structures and starting to organize themselves. Not really exactly like cortex, but at least much more organized way than the other structures. And this is from our Hermes project from Ilaria Decimo group from Italy. And left you can see the neurons on the map 2, and GFAP marks the astrocytes. So you can see some organization there, and the structure was about to mimic the hippocampus. So this is the kind of how it looks in 3D. So it is some resemblance actually. So these kind of models are also emerging. Not very much studies on stimulation yet though. This is just one example we did in collaboration with University of Auckland and University of Wollongong, Australia, and on 3D. An idea here was to build this kind of 3D electroarray that you can see on the right. This is from the Auckland site. And then try to kind of look how this electrical stimulation given through these electrodes kind of drive or boost this network on top that are made from human iPS stem cell-derived neural cells. And here we didn't look the activation or how the neurons are activated, because this is early network. So here you look actually the functional imaging, and that is how the networks start to evolve on top of these stimulated electrodes. And clearly the stimulation enhance the kind of growth of actions and enhance the network kind of construction. Just an example of kind of what you can do in vitro as well. So there's a lot of work actually done, interesting work on so-called biohybrids, where we can have kind of combination of biological systems and ICT. Of course, the whole MEA system is like that, but looking more like functional things. And this interesting paper is quite many years old, but still it's kind of interesting approach where they hooked up this MEA system, a robot with sensors, and then they put it on, and the control of the robot movement from the neural system and the maze. Very interesting story. It was a little bit still like a toy system, because there was not really learning, but it was more like selection on input and output channels from the MEA that provided this kind of logic for this, let's say, from sensors to motor. But this just shows the kind of interesting area that you can go as well. What we have done recently is kind of looking at the kind of idea that how we can have closed loop on demand based stimulation in vitro. I would have hoped to give you actually a little bit more on this, but present situation have kind of, let's say, stopped some of our experiment on lab, and this is based on our early paper that was published last year in a conference. So if you look at the MEA systems, you have the array, then you have the head state amplifier, interface board, and PC software at the end. Normally the stimulus, you decide what the stimulus is, you drive it, and then you analyze it later what happens. Actually, what is nice thing is that the interface board has a detailed signal processing that actually could offer real-time analysis. So how about to drive this interface board and build an adaptive stimulus system that stimulate only when there's some condition happening on the signaling of the neurons on the MEA system. So I have this kind of closed loop system that neurons itself kind of tell when to stimulate, or at least there's a logic behind. So what actually we did first was very kind of simple scenario. And this is really an idea that we record from one channel of the MEA, and then select another channel to stimulation. And of course, we look from the microscope imaging. There's a kind of connectivity between these, and also there's a synchronous activity between these cells. And when something happens on the recording electrode, we start to stimulate. And this is really kind of idea very simple. If there are kind of mini bursts or 3 spikes in 500 milliseconds in our observed electrode, then we start to have 5 basic pulses, 100 pulses actually on our receiving electrode. Of course, these are like the signal they look like. This is a spontaneous activity, there are spikes, and then you have how the spike look like. And when you stimulate, you can have huge signal there, because they are normal are like tens of millivolts, and then this is like a volt signal, and then you can see the response on different channels. Unfortunately, this response is not necessarily kind of straightforward, because these are really random dissociated cultures. But when you look at the statistics, something will happen. But let's look actually first what could happen also on the electrodes. So on the left, you can see the original, let's say, typical neural structures. So that the green ones again are the neuron system, and the red ones are glia cells, and blues are cellsomas. On the right, it's after stimulation. You can see that the electrode black dot on the center, they are not really green very much around it. There's green, but it's very much blended with the glia cells. So there's definitely a change after stimulation there. And this is what happens in vivo as well when you have implanted electrodes. When we now look at the average spike and counts on what happens there. So this is a baseline before stimulation. And you can see there that we use the stimulation on like 20 days or long, kind of many days stimulation as well, and then there's a post stimulation at 20 days. So this is before the stimulation, then after the stimulation. So definitely the functional stimulated cells, feedback stimulated, they are more active than the predeterminate stimulation. And also it remains on post stimulation. And same thing happens on bursting. So this is how when the neurons are these spike trains. So the functional stimulation clearly provided kind of different feedback than the normal stimulation. And of course, this is very preliminary, but it seems that the effect of stimulation nervous system depends on previous state of the system as well. And maybe, hopefully this brings something interesting in our later studies. Okay, think about future, where we go. I kind of introduced 2 of our projects we are involving. Maybe it's kind of some idea. This is our Center of Excellence in Body on Chip research. And as I said, we have 3 groups from different biology. So they represent different kind of cell types. And then we have people from kind of engineering side, micro nanosystems, biomaterials, biophysics and imaging, like myself. And what we do here is really environments for the cells to grow, but not just cells. We would like to do system that are tissues, because in a way, like neurons are not the only type of cells in the brain. There are a lot of different type of cells, and cardiomyocytes are not only cells in heart. So you have to think what is the real tissue. And then of course every tissue have innervation, every tissue have vascularization. How to build those in these in vitro systems that mimic the body. So this is what we have done already. So we have environments we can grow, we can follow them optically and electrically by these MEA systems, and control all the environment for most environmental factors on these systems. And this kind of early kind of idea prototype where we are heading. So really looking for this kind of multi-compartment chip. And idea is that there should be a common vasculature and innervation going from one chip to another. And of course, this enables not just to look at the input output of neurons, but looking like systemic effects. What is the effect of stroke? Some metabolic challenges like oxygen deprivation. What happens there? And of course, this is the idea that we would like to go is more like diversity, kind of personalized medicine. How these can be used for testing drugs or looking diseases, personalized diagnostics and drug discovery. Another project that we are involved is this kind of future emerging technology project in EU called HERMES, as I told before. An idea there is to heal the brain disorders with tissue transplant, but not just like to have the tissue, or not just like to have a neuroprosthetic like electrodes to stimulate something, but really idea is to put these together so that we actually have implanted cells, and then also this neuroprosthetic idea. And of course, it means a lot of different kind of areas to study. So we have in vitro cultures for organoids for the graph. We have in vitro and in vivo models of animal models for epilepsy. We have a lot of work in stimulation in vitro and in vivo. We have to develop new methods for assessing neural dynamics, look into artificial intelligence control, and develop this kind of implantable neuromorphic system with electronic CMOS and MEMS. But this is a future emerging technology project, so this is interesting work, and of course 4 interesting years ahead. Okay, so this is kind of concluding part, and I would like to say that these kind of in vivo, ex vivo slice in vitro, that means, has had already a lot of impact on neural research. But now these new, let's say, defined neural culturing systems are emerging, especially using human stem cell-based system. And both directions are developed now to really better control the neural system on chip, and also to improve the biometric system. And these will have potential for another direction is personalized medicine, really novel tools for systemic effects. Another direction is looking these for this bio-ICT interactions and how to make the transfer from these in vitro systems to in vivo. I'd like to thank my lab, and especially those people who are involved in electrophysiology and doing the neural work, Jarno Tanskanen and Annika Ahtiainen, especially because some of the material was here in this project, and our collaborator Susanna Narkilahti. We have done a lot of work in neural side, and our human center of excellence work there as well, and of course the Hermes consortium. And thank you for listening. I hope you enjoyed the talk and got interested also the translation from in vivo brain stimulation to in vitro. And there you can find also some of the let's say links to our work as well. Thank you for listening.
Ladies and gentlemen, let me briefly introduce myself. My name is Patrick Shimko, and I'm a PhD student at the Masaryk University in neuroscience program. Thank you for the opportunity to speak today in this online conference. And I would like to take this opportunity to talk about my research that focuses on enhancement of visual cognition in healthy seniors using transcranial direct current stimulation. Let me start with some general information about non-invasive brain stimulation, cognition, and brain networks. Research on non-invasive brain stimulation techniques alone or in combination with cognitive training has gained attention as a means of cognitive enhancement both in young and older adults. Given the decline of cognitive functions, even in healthy aging, improving cognition is particularly relevant for older adults. On the level of brain networks, aging-related suboptimal neuromodulation may hinder the information processing within and between cortical networks, and therefore the focus should be on restoring the neuromodulation using non-invasive brain stimulation. In my research, I focused on visual cognition, particularly on visual working memory. Working memory functions are subserved by complex interplay between large-scale brain networks, among which the frontoparietal networks sends top-down signal into the posterior areas, in our case, into posterior visual areas. In my study, we had two main objectives. First, our objective was to identify the optimal TDCS protocol that would have a beneficial effect on visual working memory. And the second was to use resting state functional connectivity analysis to understand the neural mechanisms underlying TDCS-induced modulation of the visual processing. In the study, we used two montages. First was the gold standard target, anodal stimulation over the left dorsolateral prefrontal cortex with the cathode placed over the right orbital frontal cortex. You can see it on the picture on the top, the pictures on the top. And the second target was more right-sided. The target for the anode was the right middle frontal gyrus with the cathode placed over the right interparietal lobules. This montage was based on the task-based activation during the visual working memory from our prior study. For more detailed information about the stimulation parameters, see the information above on the slide. Our main behavior outcome was visual object matching task that was performed prior and after the stimulation, so we call it as an offline task. The visual cognitive or the visual object matching task consisted of a sequence of pairs of emotionally neutral images of common objects, and these objects were either from a conventional viewpoint or from the unconventional viewpoint. So basically the objects were spatially rotated, and the subjects had to decide whether the objects are the same or not. The second task was performed during the stimulation, so it was the online training task, visual working memory task with faces and scenes. During this task, or during each trial in this task, participants observed sequences of two faces or two natural scenes that were presented in randomized order. The task differed in instructions informing the participants how to process the stimuli. They had either to remember faces and ignore the scenes, or they had to remember the scenes and ignore the faces. After a short delay of a period of time, few seconds, the participants had a face or a scene stimuli presented in the screen, and the participants had to decide whether the stimulus matched one of the previously seen stimuli in the sequence or not. The task had a block design. There were 6 blocks of faces and scenes. The blocks were randomized in order. In both tasks, we measured the speed of the reactions, the reaction times, and accuracy scores. And 70 females and 8 males. All the participants had no cognitive deficits. This was based on our results of detailed neurocognitive battery that was performed prior to the study entry. All the patients had no history of neurological or psychiatric disorders. The present study had a double-blinded crossover design, randomized double-blind crossover design with 5 sessions. The first session was always a training session where all the participants learned both of the tasks, how to perform the task, and went through the nerve psychological assessment. After that, 4 sessions, stimulation sessions were performed in randomized order, 2 real stimulation sessions with 2 different stimulation montages, and respective 2 sham sessions in randomized order. Within each of the sessions, the participants performed visual object matching task prior and after the stimulation, right after the stimulation, and the resting state functional and MRI scanning prior and after the stimulation. During the stimulation, all the participants performed visual working memory task with faces and scenes. This was performed because of the known synergic effect of combining TDCS with the training task. In our resting state fMRI analysis, we focused on 2 resting state networks, the visual network and the frontoparietal network. First, we used independent component analysis, and we identified components that corresponded to networks of our interest. And further, on the base of peak voxels within these networks, we selected the regions of interest for the resting state functional analysis. You can see the seeds that we choose for the frontoparietal network on the top right corner or image, and on below this image, you can see the seeds that were selected for the visual network. In this slide, you can see the results of the main behavioral outcome, the visual object matching task. The results for the left dorsolateral prefrontal cortex mortgage showed beneficial effects and accuracy scores. Here, we calculated the differences between the scores after the stimulation minus the baseline scores, and we saw and we observed significant differences between sham and real stimulation, favoring the real stimulation in overall accuracy and conventional view condition, while there was a trend to positive beneficial effect for the real stimulation in the unconventional view condition. Further, we analyzed separately the effect, the scores after versus baseline stimulation for real and sham stimulation for all the conditions. And in all of the 3 conditions, overall accuracy, unconventional view condition, and the conventional view condition, we saw only in real stimulation positive significant improvements, while in the sham stimulation there were no significant improvements. Here in the left side, you can see on the figure the changes in accuracy after the real and sham stimulation compared to the baseline. After the right-sided montage over the right middle frontal gyrus, there were no significant effects on the speed or accuracy in this offline visual object matching task. However, after we analyzed the results of the online training task that was performed during the TDCS, we saw a gradual decrease in reaction times in this right-sided montage. You can see it here. In the active condition, while during the sham condition there was a fluctuation in the speed of reaction. Specifically, in the last block, there was a significant decrease, significant difference between the real and sham stimulation in the reaction time, favoring the real stimulation group. suggesting that the right-sided montage may have positively affected the performance of the online visual working memory task. In the next step, we explore the neuronal underpinnings of the underlying TDCS-induced modulation of the visual working memory. For the left dorsolateral prefrontal cortex montage. We use linear mixed models to evaluate the effect of the factor time, stimulation, and the interaction of these two factors on the resting state functional data between the regions of interest. Within the visual network, we found a significant change between the seeds that were placed between the middle occipital gyrus and the right fusiform gyrus. There was a significant effect of interaction time and stimulation, and the pulse analysis showed a significant effect of the factor time. For the active condition, there was a significant increase in the connectivity after compared before the stimulation. In the frontoparietal network, there was a significant change between the left dorsolateral prefrontal cortex and the Goldman area 40 or the left supramarginal gyrus by the means of significant time by stimulation interaction. In conclusion, the stimulation over the left dorsolateral prefrontal cortex led to positive after effects in our main behavioral outcome in the visual working memory task accuracy. And the active TDCS over the left dorsolateral prefrontal cortex was accompanied by an increase in the network connectivity within the visual and frontoparietal network. Further, we found that the stimulation over the right middle frontal gyrus may have influenced the speed of the reaction in the online working memory task. Taken together, the frontoparietal TDCS enhanced the visual cognition in healthy older adults, while influencing the resting state connectivity within the visual and frontoparietal network. Here are my references, and thank you for your attention, and I'm looking forward for the discussion and your questions.
Good morning, my name is Massimo Bertoli and I am a PhD student from the University D'Annunzio of Chieti and the LETS laboratory of the Italian Research Council. First, I would like to thank the organizing committee for giving me the opportunity to contribute to this online conference with an oral presentation about the effects of a personalized neuromodulation against multiple sclerosis fatigue. We are interested in understanding and hopefully treating MS fatigue because it is one of the most frequent and bothersome symptoms of the disease, and at date there is no effective therapy against it, unless with the unpleasant side effects. We moved from the knowledge that MS fatigue origins from a functional rather than structural alteration, mainly involving the primary sensory motor and parietal networks, thus suggesting potential benefits from treatments that aim to compensate the neuronal electric communication dysfunctions. In particular, literature on MS fatigue indicates a specific involvement of the sensory motor network, with primary motor areas reported as excessively excitable, while S1 and primary and postparietal nodes of the somatosensory network less excitable than normal. In addition, connectivity between S1 and M1 appears to be impaired in fatigue patients and sensitive to alteration of neural networking. Based on this knowledge, our group carried out a personalized stimulation protocol called Pharemus, that is fatigue relief in multiple sclerosis. Pharemus consists in a 5-day anodal TDCS, one excitability, while trying to keep M1 unstimulated. Given the anatomical contiguity of these cortical areas, we used a personalized electrode shaped on MRI-derived individual cortical folding, properly positioned over the target areas. The efficacy of this innovative approach for relieving fatigue in MS patients with minimal clinical severity has been observed previously by two independent RCTs, crossover and sham-controlled, that achieve an efficacy in relieving fatigue with a mean effect size of 1.3, that is a very large effect, as indicated by the fatigue levels reduction expressed by the modified fatigue impact scale percentage after the real treatment, 28% in the first group, 42% in the second. Moreover, LETS laboratory implemented a novel concept procedure for the identification of neuronal sources, called functional source separation, which proved capable of resolving cortical sources topographically close to each other, like M1 and S1. Here we applied the functional source separation algorithm to extract the activity of S1 and M1 hand representation in order to study their activity through time, that is, their neurodynamics, and the effects of Pharemus on them. As you can see in this picture, we identified M1 activity of each hand during a motor task consisting in an isometric hand grip against a semi-compliant air bulb at 5% of maximum voluntarily contraction, maintained for periods of 20 seconds, here between the green and the red marks, intermingled by 10 seconds periods of rest to avoid task-induced fatigue. S1 activity instead was carried out from cortical responses to electrical stimulation delivered to the median nerve at the wrist of either left and right hand. Once disentangled the activity of M1 and S1, we were able to study their neurodynamics at rest. The experimental procedure included: at day 0 before the treatment, we performed the personalized electrode shaping based on individual MRI once for each person with MS. In blocks 1 and 2, equal but each stimulation real or sham, we collected the data at rest, pre-Pharemus, and positioned the electrode over the target areas for each TDCS stimulation. repeated for 5 days of treatment. After a period of time necessary to return to the baseline, in the 5th and last day we perform again the data collection at rest. The data collection, pre- and post-Faremus, included the fatigue levels assessment via modified fatigue impact scale and the EEG at rest for the brain effects. To assess the resting state local activity, we used the fractal dimension of the left and right primary sensory and motor cortices. We used the fractal dimension because it already proved to be a good candidate for assessing the brain network state and functionality and also for typifying cortical districts at rest. Besides, we believe that complex system measures are proper to describe the physiology of neuronal electric activity whose dynamics display hugely complex temporal structures. To assess the resting state functional connectivity between the 4 nodes of the sensory motor network, we used the mutual information. Moving to results, we found that the Faremus neuromodulation had a distinct impact on the activity of S1 and M1, with major involvement of S1, in particular of the left hemisphere. In fact, as we can see here, pre-Faremus fractal dimension of the left hemispheric S1 turned out to be greater in fatigue MS patients than controls. Faremus mostly changed fractal dimension of left S1, and the difference in before disappeared after the treatment. Of note, whereas S1 activity differed from healthy controls, this did not happen for M1. These results indicate a higher impairment of S1 with respect to M1, in line with our hypothesis that the parietal cortex mechanisms play a crucial role in generating MS fatigue, with the local impairment more evident in the dominant control region. Concerning intra- and interhemispheric functional connectivity between S1 and M1 areas, we observed that overall the mutual information values improved, mostly between the left and right M1. Pre-treatment, comparing the functional connectivity of healthy controls and MS fatigue patients, we can see that these latter had lower values. Post-treatment, the mutual information value improved significantly, reaching a level comparable of that of the healthy controls. Thus, rebalancing the sensory-motor functional connectivity results in a reinforcement of the interplay between homologous M1 areas that contributed significantly to fatigue amelioration. So, what did we learn about MS fatigue? The parietal, more than frontal cortical networks are involved with MS fatigue. Faremus neuromodulation rebalanced the sensory-motor functional connectivity, reinforcing the interplay between homologous M1 areas. The neurodynamics changed by Faremus, that is the fractal dimension and mutual information, explain together 48% of fatigue levels amelioration. Lastly, the fractal dimension among the complexity measures can quantify relevant properties at rest of the cerebral networking organization at the single node level. Thank you for your attention.
Hello everyone. I hope everybody is doing well and healthy. I'm Tiam and it's my honor to be at this conference with great scientists and audiences from all over the world. Good time to all, and I appreciate all previous presented great works and suggestions. About my background in brain stimulation, I like to say that I became familiar with brain stimulation applications after my master when I started to use for the first time TDC simultaneously with cognitive tasks for patients in brain and cognition clinic in my country, Iran, as some might know it as Persia, and we got lots of good results. That's why I'm working now in the group of Professor Nietzsche at IFADA in Dortmund, Germany as a PhD candidate. And today I will be discussing the latest finding from an EU-funded Luminous project. The focus of this research project is to develop new and novel stimulation techniques for exploration and alteration of human consciousness. And we are proud to be part of this exciting work. As a background, we all know that our brain is actively firing millions of neurons, and when we measure this activity over a period of time, we get patterns of brain oscillations. From EEG and MEG studies, we think that this oscillatory activity and synchronization might be relevant for many important cognitive functions such as motor coordination, information transfer, memory, and plasticity. At the same time, we also have some evidence that abnormal oscillations are associated with neuropathological disorders like Parkinson's disease, epilepsy, and disorder of consciousness. Since imaging studies do not give us the direct causal evidence, a recent trend in the field has been to look at using brain stimulation in order to alter oscillatory activity directly. The goal is not only to understand cognition but to treat neurological disorders. For modulating oscillatory activity, there are two main techniques which have been developed. One is RTMS, which uses high intensity electromagnetic pulses to induce neural activation and oscillatory activity, although this is short lasting. On the other hand, we have TACS. With low intensity TACS, the oscillatory activity can be amplified in the brain through entrainment, but this technique is less effective when there is no endogenous oscillation at the target frequency. This is problematic. Why? Because in many neurological and psychiatric disorders, in many of them, oscillatory activity in relevant frequency bands is diminished or completely absent. So TACS might not be very suited. Therefore, we had these questions whether it might be possible to combine and synchronize these two techniques in order to induce and stabilize any desired frequency in the brain. If we could achieve this, then it might be possible to bring these techniques to research and clinical applications. In order to combine these two techniques, we need to first develop a way for both stimulation techniques to synchronize with each other. Otherwise, the effects from one stimulation might interfere with the other. In a first step, we built an electrical circuit where RTMS pulses could be synchronized simultaneously with TACS. As you can see here, we designed a montage where TMS coil was placed directly over the top of each TACS electrode and delivered pulses in phase with TACS. And in the first set of experiment, we applied a stimulation at 10 Hz over the prefrontal cortex and measured changes in resting EEG, resting state EEG activity before and after stimulation, as you can see here. And we found that phase synchronizing RTMS pulses to the peaks of TACS sinusoids resulted in an enhanced and lasting increase in 10 Hz oscillation, which was more effective than each stimulation protocol on its own. Also, the effects was not observed when RTMS was synchronized to the throat. Given this first promising result, we were now interested to use this combined stimulation protocol for exploring causal relationship between oscillations and cognitions. In this view, we looked first at whether we could also enhance resting state prefrontal theta oscillations with our combined stimulation protocol, and secondly, whether such a prefrontal theta enhancement could also be directly linked with improved working memory. I will go through the results of each experiment one by one. In our first experiment, we look at whether phase synchronizing stimulation could enhance resting state EEG power. In this experiment, we had 25 participants who took part in 6 randomized sessions, and 6 Hz RTMS plus TACS was applied over the bilateral prefrontal cortex, and our stimulation protocol was 20 minutes continuous TACS plus 30 seconds on and off RTMS, and we recorded EEG in eyes open and eyes closed conditions at baseline, and also every 10 minutes after stimulation up to 60 minutes. In order to quantify the effects of combined stimulation, we compared our combined stimulation with other protocols, which each stimulation protocol was on its own. And as well as to a protocol completely sham. We also included 2 additional protocols for exploration. One protocol was theta gamma 80 Hz by 6 Hz cross frequency coupled TACS waveform, which has been demonstrated to be involved in working memory processing. A second protocol was to explore whether after effects could be sustained longer by intermittently continuous TACS every 5 minutes. We found that when we compare changes in 6 Hz power to sham stimulation over the 60 minutes time course of the session, all 3 combined protocols showed a significant enhancement in power in both eyes open and eyes closed condition. The grand average change in 6 Hz power for 60 minutes shows that in both eyes open and eyes closed conditions, the 3 combined protocols showed significantly enhanced 6 Hz power relative to each protocol on its own and to sham. When we look at changes in 6 Hz activity over the whole brain, we found a pattern of enhanced power in both frontal and parietal region. In order to assess whether these changes were functionally coupled, we measured changes in the functional connectivity between these two regions. And functional coupling was enhanced with the three combined protocols relative to sham stimulation in both eyes open and eyes closed condition. So the conclusion from our first experiment was that we were able to enhance 6 Hz activity over the prefrontal cortex by phase synchronized RTMS and TACS with effects lasting up to 60 minutes after stimulation. Moreover, we also found the network effects in the frontoparietal regions, which suggests that these stimulation techniques might be useful for altering network activity. With these positive findings, we then asked the question of whether these techniques might also be effective for functional outcomes in cognition. Specifically, we thought about working memory, since it's shown to be directly related to theta activity. So we in the second follow-up study we conducted the same stimulation, but this time we asked the participant to complete the 3-back letter task before and after the stimulation. When we computed the results, we found that both the combined RTMS plus TACS protocol, as well as the RTMS plus TACS theta gamma protocol, significantly improved overall performance accuracy on 3-back. On a 3-back task as measured by D prime statistics when compared to sham stimulation. We also found a non-significant trend where the combined RTMS plus TACS protocols also decrease reaction time, meaning that subjects did not only perform more accurately but also slightly faster. So to conclude, we are able to demonstrate that these combined techniques also result in functional changes to cognitive process like working memory. Specifically, we found that RTMS plus TACS over the bilateral prefrontal cortex with both 6 Hz TACS and also cross-frequency 6 and 80 Hz TACS protocol could enhance working memory performance, although there were no significant difference between these two protocols. These findings are still in their early stage, and we expected to carry out more work in this line, like studying the mechanism behind the after effect and whether they are depends on synaptic mechanism, and also we are interested to experiment with the stimulation parameters in order to further optimize the protocols. Taken together, we believe that this phase synchronized oscillatory stimulation could be a viable approach in cognitive neuroscience for directly exploring causality between cognition and oscillatory activity, and also as a therapeutic approach for clinical cases of abnormal oscillation, like in epilepsy, major depression, or disorder of consciousness. With this slide, I want to thank all my colleagues, friends, and family who helped me in and support me in this project. And this is my email, and I am open to any idea or collaboration. And if there is any question or comment, I will be happy to hear that. Thanks for your attention.
Good morning, everybody. I'm Erika Baroli from the Mediterranean Institute for Transplantation and Advanced Specialized Therapies in Palermo, Italy. And I will present you my work titled TMS-EG, a promising tool to study the TDCS effect on cortical excitability. So the aim of the project is to explore the mechanisms underlying the effect of cathodal TDCS. TDCS in general is a non-invasive brain stimulation technique nowadays increasingly used not only for scientific purposes but also to treat and rehab many different neurological and neuropsychiatric disorders. However, the neurophysiological mechanisms underlying its effects are still unclear, and this is especially true for cathodal TDCS. So, in our opinion, a complete understanding of this mechanism will be crucial to better define the TDCS clinical use. To do that, we decide to use the TMS-EG integrated system because this specific technique is able to maximize the advantages in using TMS and EEG. Because thanks to EEG, we were able to collect real-time and direct information on cortical excitability. And combining EEG with TMS, we were able to explore the cortical activity also of that areas otherwise not functionally measurable. So, to reach the aim of the project, we starting from the results of a previous study focused on anodal TDCS. In this experiment, Romero Laura and colleagues observed a rise of cortical excitability in a wide spread of cortical regions. So here, in these studies, we performed two different experiments. The first one in which our participants were at rest during TDCS stimulation. In the second experiment, our participants were involved in a task during TDCS stimulation. So for both the experiment, we used the same TDCS parameters used previously in the anodal TDCS study. And in particular, you can see on the left side of the slide the position of the cathode and the anode. And in particular, the cathode was put on the right posterior parietal cortex, while the anode was put on the left supraorbital contralateral regions. On the other side of the slide, you can see a modelic reconstruction of the electric field induced by this specific montage and these specific parameters. So this is the experimental setting in which we try to combine TMS-EG system with the TDCS stimulation, and in particular here on the right side you can see how physically we try to do that. We put the TDCS patch under the EEG cap and we had to remove the EEG electrode over the TDCS patch. And on the left side you can see a 3D reconstruction on the individual MRI in which you can see the TMS hotspot that was the left posterior parietal cortex. The green circle that you see here is the electric field induced by the TMS. And the blue square is the reconstruction of the TDCS patch in dimension and position on the right posterior parietal cortex. So, starting from the first experiment, this is the experimental design. We collect TMS pulses around 180 TMS pulses 3 times before, during, and after TDCS stimulation. And we computed global and local index of cortical excitability in 3 different time windows, starting from 0 to 150 milliseconds after the end of TMS pulse. At a global level, we computed the signal deriving from all the 60 channels, EEG channels. And at a sensor level, we didn't found any significant differences between the 3 recording sessions in all the 3 time windows. At a local level, we computed the signal deriving from 4 different clusters of 4 electrodes each. 2 frontal clusters, one on the left and one on the right side of the brain, and 2 parietal clusters corresponding to the TMS and TDCS hotspot. So also at a local level, we didn't found any significant differences in the cortical excitability. The absence of significant differences was also confirmed and observed at a sources level, where again we didn't found any significant differences. So comparing the results of cathodal TDCS with the previous one on anodal TDCS, we can see a sort of dissociation, because anodal TDCS seems to be able to induce an increase of cortical excitability also during and after TDCS stimulation in the 2 earlier time windows. So the first conclusions seems to demonstrate that cathodal TDCS is not able to modulate the cortical excitability neither online nor offline. However, recently study on computational model and animal models seems to observe that TDCS effects may depend on the level of basal activity in the system. And if it is true, cathodal to observe some changes in cortical excitability induced by cathodal TDCS, it could be possible that we need to stimulate a system with a higher level of basal activity. So to do that, we decide to pre-activate the target areas with a cognitive task. So we move through the second experiment, in which our participants during TDCS stimulation were involved in a task. And in particular, this is our experimental design. We collect TMS EEG data before and after TDCS stimulation, because during TDCS stimulation, our participants performed the Posner task, that is a visuospatial orienting attention task. The effectiveness of the cathodal TDCS stimulation on the performance in this task was previously tested in a pilot study. At a behavioral level, we fortunately confirmed the results observed in the pilot study, and in particular, we observed an increase in reaction time during cathodal TDCS stimulation compared to the sham session. At a neurophysiological level, we perform a cluster analysis that show differences between the 2 recording sessions in a clusters of electrodes that cover the areas strictly involved in the task performance, and in a later time windows that follow between 180 to 230 milliseconds after the TMS pulse. The same significant cluster was also observed at a sources level, where we confirm the results observed at the sensors. So The conclusion of the second study seems to demonstrate that cathodal TDCS apply on right posterior parietal cortex is able to affect performance, and in particular increasing reaction time. The second conclusion of the second study is that the cathodal TDCS seems to be able to reduce cortical excitability in a later time windows compared to anodal TDCS stimulation and in areas strictly involved in the task performance. So, taken together, our results seems to demonstrate a sort of non-linear effects induced by anodal and cathodal TDCS. And this is especially true when we move from sensory motor domains to high level of cognitive function. Our results also seems to corroborate the results obtained in the computational and animal model study, in which the effect of cathodal TDCS and TDCS in general seems to depend on the level of basal activity in the cortex. And because in our study, we stimulate the same target area with the same parameters, but also when the areas was already activated by cognitive task, we were able to observe some changes in cortical excitability induced by cathodal TDCS. And the last but not least point is that the modulation of cortical excitability could be the neurophysiological mechanisms underlying the behavioral effects, because the modulation of cortical excitability that we observed also with cathodal TDCS were strictly related to that areas involved in the task. So that's all. Before I finished, I want to thank all the people that significantly contribute to this work, and obviously I want to thank you all for your attention.
Hi everyone, my name is Maats Wisniewski. I'm from the Donders University in Nijmegen in the Netherlands. And in the last 3 years we've done 2 exciting experiments in the lab which you see behind me. And today I want to talk about that in this little presentation. So yeah, let's get started. So in the study we tried to influence reversal learning performance by using transcranial alternating current stimulation. And I presume that most of you are familiar with TACS, but just to quickly reiterate, with TACS we use an electrical stimulator to induce some oscillations on the scalp, which then are thought to entrain oscillations within the brain. So for example, if you apply oscillations on the scalp in a theta range, then you would expect that theta oscillations within the brain are enhanced. And the same is true for any other stimulation frequency, for example, the beta frequency. So in this study, we wanted to use those the TACS to have an effect on reversal learning. So people in our study received a task in which they saw 2 numbers, as you can see in the left upper corner. And those 2 numbers represented points that they could either win or lose. So when they choose the 25 option, they could either win 25 points or lose 25 points. Or when they choose the 5, they could either win 5 points or lose 5 points. So basically those reflect a high risk and a low risk option, because with the higher number you can win a lot, but you can also lose a lot. And with the lower number you can win just a little bit, but also lose just a little bit. And our task, as you see in the left lower part of the screen, you see that we designed the task in such a way that in the beginning the high risk option was the better option in 80% of times, and only in 20% of times the low risk option would be better. And over time people should learn this, and then after a while this switches to the lower option being the better option, and then after a while it switches back to the high risk option being the better option. And designing the task in such a way allows for looking at learning in two different ways. So first you can look at the learning speed, which is basically the number of trials needed for people to reverse their strategy from a high risk strategy to a low risk strategy and back. And second, the rule implementation, which basically is how well they implement the rules. So if the high risk option is the better option, are they actually taking the high risk? And if the low risk option is the better option, are they actually taking the low risk? Or are they hovering somewhere in between and not really know what they're doing? So we wanted to look at those 2 aspects of learning. And also we wanted to have an idea what happens to the brain when we stimulate with TACS. And that's why we also recorded a resting state EEG before and after the task. Ideally you would want to do this during the experiment, but looking at EEG while applying TACS is very, very difficult to say the least, and we were not able to do that, so that's why we didn't. So what did we do? We applied theta. In the first experiment we applied theta oscillations, because we know that theta oscillations are important for reward driven motivation. So when there is a learning task like this where there's points involved, you would expect that theta oscillations are important for that. We know that theta oscillations are reflected by long-range cortical to subcortical interactions, and also interactions between the left and right hemisphere, and that's why we stimulated left and right frontal cortex. We used 1 milliampere and stimulated during the entire task, which was around 12 minutes. So what did we find? So we tested a total of 50 participants, which is 25 per group, because we had 1 active group and 1 sham group in which people didn't receive any stimulation. So the dots represent basically the average risk-taking participants took, and as you can see, over time in the first 20 trials people learned that the high-risk option is the better option. And then from trial 40 onwards you see that people tend to go to the low-risk option, and that's correct because that's when it reverses to the lower risk option. And the lines represent basically how fast people do this. So what you can see is that there is a small but significant difference in how fast people learn the switch from high risk to low risk. The people who received the real theta TACS, they needed less trials to reverse from high risk to low risk compared to the people who received the sham simulation. In the second reversal from low risk to high risk, we see the same trend, with people who receiving the real theta TACS being a little bit faster. However, this was not significant. So you would say, okay, this is cool. Theta TACS is good for reversal learning performance. However, when we look at the very last block, we see that people who received the real theta TACS, they didn't use as much high risk as the people who received the sham simulation. And that is interesting because in the last block, you should take the high risk. That means basically that the rule implementation, basically the rule to use a lot of high risk, wasn't that well implemented at the end by the people who received real stimulation. So although theta TACS is better for or improves learning speed, it's not necessarily good for the rule implementation. So what happened when we look at the EEG results? We first looked at coherence measurements between left and right hemisphere, but we didn't find anything. I don't even have a graph of it because it's just very, very sad to see almost. So that's why we looked at the power. And what you see here in this graph is the change from pre to post difference between the real and the sham theta TACS. And what you may expect is that theta TACS increases theta power in the frontal cortex. This didn't seem to be the case. If anything, there was the opposite. If you look at the white bar in the left hemisphere, there was, if anything, a decrease in theta power. However, it was not significant, so we should conclude that there was no difference. Also, there was no difference in the right hemisphere. However, my supervisor suggested me to look at the theta-beta ratio, and that is because in research with ADHD patients, the theta-beta ratio is often used as a measure to look at cognitive control and cognitive performance. And what we see in patients with ADHD is that they tend to have higher theta-beta ratios, so more theta oscillations compared to beta oscillations, and this does reflect poorer performance in reversal learning tasks, where they often persevere and stay with their first strategy and never really implement the new rule. So analogously, you would expect that a lower theta-beta ratio would be associated with better reversal learning performance, and that was the case here. Here we saw that the theta-beta ratio, at least in the left hemisphere, was decreased from pre to post in the real TACS conditions. So, of course, these resting state measurements were done after the task ended and after stimulation ended, so whether this really reflects what happened during the task is a little bit hard to say, but at least it does seem to make sense. It does seem to make sense that a lower theta-beta ratio is coupled to better reversal learning performance. All right, so after that we thought, okay, theta-beta ratio, that's interesting, but there is a beta in theta-beta, so also we should have a look at the beta oscillations. So we thought, why not do the exactly same experiment, but this time with beta oscillations? We tried to replicate the study completely. However, we used a slightly different montage in which we used an in-phase and an out-of-phase montage, basically to have one hemisphere in the same phase or in the opposite phase. However, I should say that since we didn't really focus on two separate areas, I'm not so sure that that this really made sense afterwards. At the time, it seemed like a good idea, but afterwards, I'm not so sure that this was the best way to look at it. In the results, you will also see that that there's no real difference between the two conditions. So yeah, I'm not going to go into too much detail about that. Now, of course, if you have any questions, you can ask me afterwards. Another thing is that we used some LED lights next to the eyes, which were flickering to control for phosphines, which people often observe during beta stimulation. And we also used those during the sham stimulation, so that the flickering of the lights would be the same between the sham and real active conditions. So what we found in the three conditions is that reversal learning speed wasn't different between any of the conditions. So active TACS, whether it was the in-phase or the out-of-phase one, wasn't faster or didn't produce faster reversal learning than in the sham condition. However, what we did see is that people who received active TACS implemented the rule better, particularly the rule of taking low risk in the low risk block. And also what you can see is that there's no real big difference between the two TACS conditions. And as I said, I'm not so sure that it was so necessary to have those two conditions. We probably could have just used the same montage as in the first experiment, but yeah, that was just an idea I had back then. So in this case, we find no improvement on reversal learning speed, but we do find improvements on rule implementation. So how did the EEG look like? So in the EEG, we find that from pre to post, the theta-beta ratio went up in the sham condition. However, this was not the case for the active TACS conditions. So basically what you could say is that in the active TACS conditions, theta-beta ratio was lower after the task compared to sham. However, again, this is a measurement after the task, not during the task, so whether this is a good representation of what happened during the task is a little bit hard to say. But again, it seemed that better reversal learning performance was associated with decreased theta-beta ratio, which at least makes sense. So what can we conclude? Well, we can conclude that theta-TACS improves reversal learning speed, but doesn't really help with rule implementation, whereas for beta-TACS, the opposite was found. Reversal learning speed wasn't affected, but rule implementation seemed to be better. So this suggests that those two oscillations have two different functions in learning, in reversal learning in the frontal cortex. However, interestingly, in both cases, the theta-beta ratio went down, which it's very interesting. It does make sense if you look at the analogy of the ADHD patients, but it is quite interesting to figure out in future research how this theta-beta ratio is actually related to reversal learning, and particularly those two different aspects of learning. So yeah, that is it. I hope you enjoyed the talk. If you have any questions, please let me know. And of course, you can always send me an email or ask me in the discussion afterwards. And I hope you enjoyed it. I think this is a cool research that prompts some interesting future research. And yeah, with that, I hope you enjoy the rest of the conference. Thank you.
Hello, my name is Desmond Agboada, and I'll be presenting you my research today, titled Induction of LTP-like Plasticity in the Primary Motor Cortex with Repeated Another TDCS. TDCS is a non-invasive brain stimulation technique that induces polarity-dependent after effects, and in the primary motor cortex, there is an enhancement of cortical excitability with another TDCS and a diminution of cortical excitability with cathodal TDCS. It has been shown in one of the earliest TDCS studies that the after effects of TDCS can outlast the duration of stimulation. And with the right combination of parameters, TDCS can induce LTP-like plasticity in the brain. Recently, we systematically evaluated and titrated the parameter space of a nodal TDCS with respect to stimulation intensity and duration. So we compared the 3 stimulation intensities and 3 durations with a sham to find the best stimulation protocol among this pool of protocols. And we found out that the intensified protocol of 3 mA per 20 minutes was the best among this pool. However, the after effects we observed were mostly still in the early LTP phase. So the time range was still a few hours post-stimulation. It's been suggested that to induce LTP, we will have to stimulate for a number of times within a specific interval. So space stimulation protocols have been shown to be superior to single stimulation protocols. This was shown back in the 60s with DC stimulation of the rat cortex. Binman and colleagues showed that application of 2 stimulation within a critical time interval increase the spontaneous firing in the rat cortex. To the best of my knowledge, this space stimulation technique was tried with TDCS in the motor cortex for the first time in the Montesilva study in 2013. Here they showed that stimulation with 1 milliampere in current intensity with a duration of 30 minutes enhanced cortical excitability for up to 60 minutes, which represents an early phase LTP-like plasticity. When the duration was doubled from 30 minutes to 26 minutes, there was a reversal of the polarity of after effects from an enhancement to a diminution. When a short interval was applied between the first and second stimulation, there was an enhancement of after effects, which lasted up to the next day evening. And here they used 2 different time frames of intervals, to 3 minutes or 20 minutes, which are both short intervals in this research. So they were able to induce LTP-like plasticity with a short interval. But when the interval was increased to 3 hours or 24 hours, there was no enhancement of cortical excitability, which shows clearly that to induce a late phase LTP-like plasticity, the interval must be relatively short. So we built upon this by designing a study similar to that of Montesilva, by trying to see if we can induce LTP-like plasticity with our recently developed intensified protocol, and we compare this with a standard another TDCS protocol of 1 milliamp per 15 minutes. We measured baseline excitability in the motor cortex with single pulse TMS-induced MEPs, and then we applied TDCS based on our design. So there's single session, and then there's 2 repeated sessions with a short interval repetition of 20 minutes or a long interval repetition of 3 hours. We measured after effects immediately after the stimulation every 5 minutes until 30 minutes, every 30 minutes until 2 hours, and then same day evening, next morning, next noon, and next evening. So the results showed that both the intensified protocol of 3 milliamp per 20 minutes and the standard protocol of 1 milliamp per 15 minutes enhance cortical excitability for half an hour. On the vertical axis, you can see the normalized MEPs, and then on horizontal axis, we epoch the duration of the after effects into 3 epochs. So 0 to 30 minutes, 60 to 120 minutes, same day evening to next day evening, to account for variability within the single time points post-stimulation. Also, as you can see over here, even though both enhance cortical excitability within half an hour, the intensified protocol was slightly better. And we can see here that based on this time range, we could say that these two protocols were inducing early phase LTP-like plasticity. For the repeated session, the short interval repetition showed an enhancement of cortical excitability that lasted up to the next day evening for the standard protocol. So the standard protocol here was inducing late phase LTP-like plasticity. But for the intensified protocol, the after effects lasted only up to 2 hours, which is still in the range of early phase LTP-like plasticity. For the long interval repetition, both the standard and intensified protocol did not induce late phase LTP-like plasticity. The standard protocol increased cortical excitability slightly, whereas the intensified protocol showed a diminution of cortical excitability. The asterisk is a difference between the standard and the intensified protocols. So we propose some mechanisms for this observation. First, we suggested that based on the time frames, early single session another TDCS was inducing early phase LTP-like plasticity, whereas the short repetition interval induced late phase LTP-like plasticity for the standard but not for the intensified protocol. And this we suggested could be based on two plausible reasons. First, we suggested that induction of late phase LTP-like plasticity could be due to a synaptic tagging and capture. So according to this hypothesis, silent synapses are able to capture synaptic inducing proteins or synaptic tags from previously activated synapses and are able to use these captured tags to induce plasticity at their own synapses. But the half-life of these synaptic tags is 30 minutes. So we suggested that it could be that the short interval between the stimulation was efficient to recruit these synaptic tags for LLTP induction. And the reason why we could not find the intensified protocol inducing late phase effects could be because of calcium overflow. It's already been suggested that a higher intensity of stimulation produces more calcium compared to a lower intensity. So with more calcium at the synapse, it could be that this excess calcium was opening potassium channels and thereby causing a counterbalancing effect on plasticity induction. So in conclusion, we found that single session stimulation for both the standard and intensified protocols induce only ELTP-like plasticity. Repeated stimulation with a short interval resulted in LLTP-like plasticity for the standard protocol, but not for the intensified protocol. The longer repetition interval of 3 hours did not induce late phase plasticity for both the standard and intensified protocols. And this clearly shows that to use space stimulation protocols in the motor cortex, relatively short interval is preferable to the long intervals of repetition. And it also shows that when you're using space stimulation protocols, moderately strong protocols might be superior. So this paper has already been published and is now available online. I would like to thank my supervisor, Professor Michael Nietzsche, and then my colleagues in our lab here in Dortmund. Thank you so much for listening to this presentation, and I'll be happy to hear all your questions and your emails. Thank you very much.
Hi everyone, I'm Alessia Gallucci. I'm a PhD candidate of the Neuroscience program at the University of Milano Bicocca. I'm really glad to join the Brain Stimulation Conference today. I'm really thankful to the conference board to give me the chance to present you today a very interesting social neuroscience study in which we applied the transcranial direct current stimulation to modulate the frustration-aggression link. Indeed, since the last century, a lot of scholars concentrate their efforts in shedding light on the cognitive mechanism which underlie the link between frustration conditions that are conditions or factors which prevent us to reach our personal and life goals, and the following aggressive reactions. In particular, social psychologists were so attracted by such cognitive mechanism because, as you know, while aggression was considered important and adaptive in a distant past, today it seems maladaptive and highly costly for societies and individuals. However, the brain mechanism and the neural underpinnings of the frustration-aggression link were poorly investigated by the previous literature and are still unknown. Of course, in general, what we can suppose is that the frustration-aggression link could recreate the same brain structure and areas which are generally involved in emotion regulation. A lot of neuroscience models in this field suggested that a successful emotion regulation is based on a successful interplay between prefrontal cortices and subcortical regions. In particular, scholars and a lot of research results show that the prefrontal cortices, and in particular the ventrolateral and the dorsolateral prefrontal cortices, exert a top-down control and regulation on the limbic system. As you can see here, several results from research in this field show that both the right and the left prefrontal cortex are involved in such kind of top-down control of negative emotion and aggressive reaction. In particular, a group of scholars guided by Hermon Jones in 2003 proposed a theory, a model, according to which there could be a functional asymmetry at the frontal level. In particular, this group of scholars suggested that while the right prefrontal cortex is more involved in avoidance motivational processes, as you can see at the top of this slide, the left prefrontal cortex, as you can see at the bottom of this presentation, seems more involved in approach motivational processes like anger and behavioral aggression. Such findings were very important because they opened the way for the possibility of applying non-invasive brain stimulation techniques, and in particular the transcranial direct current stimulation to modulate aggression and aggression regulation. In particular, a series of studies by Riva and colleagues show that a nodal TDCS over the right ventrolateral prefrontal cortex significantly reduced aggression caused by social exclusion or exposure to violent video games. Opposite results were obtained by Hortensius, who applied a nodal TDCS over the left dorsolateral prefrontal cortex, showing an increase in aggression as participants tending to choose louder and longer noise blast for their dummy player in aggression-inducing paradigm. Such results were very interesting and important also because they support other neuroscience model of emotion regulation. For example, the model proposed by Hoschner and Gross in 2007, according to which there are two main brain systems underlying emotion regulation. A ventral system, which recreates in particular the ventrolateral prefrontal cortex, which has a role in automatic and implicit emotion regulation, and a dorsal system, which recreate in particular the dorsolateral prefrontal cortex, which is involved in effortful and willful emotion regulation, like the one which characterize, for example, emotion reprisal strategies and procedure. Therefore, we decided to build an TDCS experiment with the aim to test the application of this non-invasive brain stimulation technique to modulate aggression induced by a frustration condition. We had two main hypotheses. The first hypothesis was that the anodal stimulation of the right ventrolateral prefrontal cortex could decrease aggression. On the other hand, the second hypothesis was that the anodal stimulation of the left ventrolateral prefrontal cortex could increase aggression. We also try to explore possible gender differences in the effect of TDCS on frustration-induced aggression. In particular, in our experiment, we involved 90 healthy participants, 45 males and 45 females, in a double-blind sham control study. First, participants received anodal or sham TDCS stimulation over the right or left ventrolateral prefrontal cortex. Then, 5 minutes before the end of the stimulation, participants took part in a frustration task, which required them to complete a series of impossible numerical sequences chosen by a dummy player. Then participants' aggressive reaction were measured by a series of well-validated behavioral tasks. For example, the competitive reaction time task, which was similar to the one I already described for the study by Hortensius, which required participants to choose intensity and duration of noise blast for the dummy player. Then a sequence chosen task and a tangram task, which require participants to choose easy, medium, or hard numerical sequences or puzzles for the dummy player to solve. For our analysis, we first computed a principal component factorial analysis to obtain a single factor of aggression, starting from our three main dependent variables. Such a single factor was then entered as a dependent variable in a between subject analysis of variance. What we found was first a main effect of stimulation. Indeed, we showed that both the right and the left side stimulation, but in particular the anodal stimulation of the left ventrolateral prefrontal cortex, significantly increased aggression. Then we show an effect of gender. Indeed, we found that in the sham condition, the males were more aggressive than females. However, we also found that such gender difference disappear after another stimulation. Indeed, we found that females who received both right and left ventrolateral prefrontal cortex stimulation were not less aggressive as males. Here, I just report the details of the analysis considering each dependent variable, and in particular the competitive reaction time task and the sequence chosen task, where you can see the significant gender by stimulation interaction effect. I didn't report the results for the tangram task because the results of this analysis was not so strongly significant. Therefore, our study confirmed previous findings by Hortensius by showing that another stimulation of the left prefrontal cortex increased aggression. However, we did not replicate the study by Riva and colleagues because we did not show that another stimulation of the right ventrolateral prefrontal cortex decreased aggression. One possibility is that the functional asymmetry, the frontal functional asymmetry I described, I already mentioned it at the beginning of my presentation, could underline such inconsistent results. Indeed, as you know, the TDCS effect strongly depend on the resistant activity of the stimulating area. And according to Herman John's model, while the avoidance processes are more related to the right prefrontal activity, the approach motivational processes like anger and behavioral aggression are more related to the left prefrontal cortex activity. And in our experimental study, we first frustrated our participants and then asked them to behave aggressively. Therefore, it could be that compared to the previous studies in our experiment, another stimulation of the left prefrontal cortex could have maximized the higher activation of the left ventrolateral prefrontal cortex. Another possibility is that the frustration could represent a special trigger of aggression, and that frustration could be different from the other aggression trigger studied by previous literature, such as social exclusion or exposure to violent video games. We confirm the well-known results about the gender differences in aggressive reaction. Indeed, we show that males are more aggressive than females in our sham condition, but we also show for the first time that such gender difference seem disappear after anodal stimulation of the right and the left side stimulation. One hypothesis that TDCS leads females to a more utilitarian behavior, but we have considered also a possible ceiling effect for males who were found to be much more aggressive than females in the sham condition. We also have to consider possible individual differences in the TDCS effects. In conclusion, our study and experiment open several future directions, and from my point of view, future studies should further investigate the neural underpinnings of emotional regulation in general, but in particular of the frustration-induced aggression. For example, future study could include a non-frustration control condition or the stimulation of a control area, which we did not consider in our experiment and protocol. For example, it could be interesting to test the stimulation of posterior cortices, such as the temporoparietal junction, which is in general involved in in-group and out-group processes, or also in mentalizing system. It could be interesting also to test other TDCS protocols, such as cathodal stimulation, or testing other focal techniques, for example, the transcranial magnetic stimulation. I would like to really thank you for the attention to my presentation, and also I'd like to thank my co-authors for their work, Paolo Riva, Eleonor Romero Lauro from the University of Milano Bicocca, and Brad Bushman from the Ohio State University. Thank you.
Good afternoon. My name is Tobias Reichenbach. First of all, I would like to thank the organizers for organizing this virtual conference. I think it's a very exciting opportunity. So my group works on hearing, and recently we have begun to use transcranial current stimulation as a tool to investigate the neural mechanisms that are involved in sound processing. And today I want to share some of our recent findings with you. And to do that, I have prepared a short presentation that I want to show you in the following. It's been a while, probably for all of us, that we've been to a busy restaurant or pub, but I'm sure we still remember that these can be very noisy circumstances where it can be quite difficult to focus on a particular speaker due to all the surrounding distractor speakers. However, for healthy adults, it's actually relatively straightforward to tune out background noise. We are really very good at it, and to kind of keep talking to a particular person. But how does it actually work? How does the brain do it? That's what my group is working on. And to understand that, we might want to take a closer look at speech and what the building blocks actually are. So the shortest time scale, about 20 milliseconds, we have phonemes. So that time scale of 20 milliseconds corresponds roughly to the gamma range, 30 to 50 hertz, the lower gamma range. Then we have syllables that last a bit longer, like 50 to 100 milliseconds. That corresponds to the theta range. And then we have a rhythm set by words. These last 20 to 300 milliseconds. And that lies in the delta range, about 1 to 4 hertz. Now, a lot of research has shown that there is cortical activity in these different frequency ranges. And this activity on these oscillations entrain to the rhythm in speech that is set by phonemes, syllables, and words. And that seems to constitute mechanisms for speech processing. We still don't understand too well how exactly that works. There's a computational model developed by Alexander Hürfield a few years ago that works with a theta module that generates theta oscillations and the second module that generates gamma oscillations. And in this model, both modules receive sound input. The theta module then acts to parse speech into syllables, and that controls the output of the gamma module, and that then encodes the syllable identities. Transcranial alternating current stimulation, as we know, can modulate activity in the brain at different frequencies. Typically, people use frequencies between 1 to 50 hertz. The currents are very small, but in animal experiments, we nonetheless find that these small currents still are sufficient to modulate this oscillatory activity. Accordingly, we might speculate that transcranial alternating current stimulation might influence speech comprehension. And in 2018, there have indeed been a series of papers published on exactly this question. What the authors did is to present people with speech and noise. And at the same time, they also stimulated their auditory cortices using current stimulation. And the current stimulation was not just alternating at a single frequency, but it followed the speech envelope. So in other words, it had a broader frequency spectrum. The speech envelope in particular mimics the rhythm of words and syllables. So in particular, it encompasses the delta and theta frequencies that correspond to that rhythm set by words and syllables. By doing so, they found that this current stimulation did indeed influence the comprehension of speech and noise by a few percent, or maybe up to 1 dB in the sentence reception threshold. What we wondered then is, where exactly does that effect come from? I just said that the speech envelope contains frequencies both in the delta range and in the theta range. You can see that in panel C in a spectrum from a typical segment of speech. But these two frequency bands appear to play quite different roles in speech processing. In particular, the theta band appears to be more responsible for syllable parsing. The delta band follows more the word rate. Some people think it's related to higher level linguistic comprehension. So we wanted to investigate this question using current stimulation. We therefore presented people with sentences in noise. We also stimulated their temporal lobes using transcranial currents. And we used waveforms that were derived from the speech envelope, but filtered to lie either in the delta band or in the theta band. And so this gives the black waveforms that you see in panels D and E. So D is the delta band portion of the speech envelope, E is the theta band portion. Clearly, the theta band portion is faster because it's a faster timescale. What we then also did is to consider 6 different phase shifts. So we shifted the phase of these waveforms by 60 degree, 120 degree, 180 degree, 300 degree, and then 360 being back to the original, meaning no phase shift. And so these waveforms are shown in these thinner lines with these rainbow colors, and together they then fill out that space. We then measured the speech comprehension in percent at a fixed signal-to-noise ratio. That meant the speech comprehension was around 50 to 60%. And we looked at how that changed potentially due to the current stimulation at particular phases. Because we looked at phase shifts, we could use circular statistics to analyze the results. We then find that for the delta stimulation, there's no significant effect of the phase. It looks like at 60 degrees, it's a little bit less, but it turns out it's actually insignificant. It's different for the theta band. For the theta band, we find that there's a significant modulation of speech and noise comprehension, in particular at the phase of 0 degree. Speech comprehension is maximal at around 60%, and at a phase of 180 to 240 degree, it's minimal at around 55%. We can fit a sinusoidal function to that, and we see that that fits the mean values shown with these circles indeed very well. So this is really interesting for the speech processing perspective because it clearly shows that the alternating current stimulation acts on the theta band portion, but not on the delta band portion of the neural machinery. We then also looked at the issue of subject-to-subject variability. So previous work had suggested that the effects of the current stimulation on speech comprehension might be quite different from one subject to another one. And we looked at that in our data, and to do that, we looked for every subject what was the best phase, meaning at what phase did the subject have the highest speech comprehension. So we call this the best phase, or B, in panel A and B. And we then measured the phases relative to this best phase. That's why we then have the best phase plus 60 degree, plus 120 degree, and so on. Interestingly enough, then, both for the delta band and for the theta band portion, we find that there's no significant effect of the phase anymore. Now, for the delta band, that's not surprising because there was no significant effect before either. For the theta band, it's a bit more surprising because we had a significant effect that now has disappeared. So what that suggests is that this fact of choosing the best phase actually makes the data noisier rather than less noisy. And that suggests that the best phase is probably actually relatively consistent across subjects. To look at it from a different angle, we then looked at the distribution of the best phases across the subjects in a histogram that's shown in panels C and D. For the delta phase, we find that most people have a best phase of 180 degree, very few a best phase at 60 degree, but actually the effects are not significant. It's not significantly different from a uniform distribution. Again, that's not surprising because there's no significant effect that we observed so far. So also it's not surprising that there's no significant distribution of these best phases. Again, it's very different for the theta portion. There we find that most people have a best phase of 0 degree, and that effect or that peak is highly significant. It's very different from a uniform distribution. So that shows what we said before, namely that there's a consistent best phase across subjects, and that best phase is 0 degree, meaning no phase shift between the current waveform and the speech envelope. That's when people do best. We also looked at if the stimulation using the theta band and no phase shift can actually enhance speech comprehension beyond charm stimulation. So far, we always compared different phases to each other, and we showed for the theta band that yes, there is an impact of the phase, but that doesn't mean that you actually improve. It could just mean it's always getting worse due to the stimulation. But this data that I show on this slide actually shows that if we stimulate with a theta band portion of the speech envelope without a phase shift, then actually we improve significantly compared to sham stimulation, and also significantly as compared to if we take an unrelated speech envelope in either the delta or the theta band portion. So that shows that not just can we influence speech comprehension using the current stimulation, but we can actually improve it. And that might have implications for treating hearing impairments, for instance. Finally, we looked at a second parameter that is important for the current stimulation, namely at the temporal delay, meaning the delay between the current waveform and the speech signal. For that, we used a range of delays going from minus 430 milliseconds, meaning the current waveform precedes the audio signal, to plus 400 milliseconds, meaning there's quite a delay between the current and the audio signal. And we use the delays based on the autocorrelation of the speech envelope to pick out maxima and minima, with the idea to get contrast between different temporal delays. What we found is that very consistently across subjects, almost all subjects had the best delay at 0 milliseconds, meaning for almost all subjects, no delay gave the largest speech comprehension. And if you look then at the actual comprehension scores, again, we find that at no delay and also at the delay of 90 milliseconds, we find a significantly larger comprehension as compared to sham stimulation. Again, going back to what we just said, we can actually improve speech comprehension by using the theta band portion of the speech envelope and having no delay and no phase shift. I want to conclude by just mentioning that we are also working on a computational model of these effects. This builds on the work by Alexander Hühnerfjell that I mentioned before on the spiking neural networks using a theta module and a gamma module. And we now use that to simulate speech and noise comprehension. And the results are quite similar in some ways to human performance. And now we also use it to simulate current stimulation. And I can hopefully update you on these results in the meeting next year. With that, I want to conclude by summarizing the main points for our findings. Importantly, we found that transcranial alternating current stimulation with the speech envelope can actually enhance speech comprehension and noise. The effects result from the theta band portion, but not from the delta band portion of the speech envelope. And the enhancement is largest when there's no phase shift and also when there's no time delay between the current signal and the speech stimulus. Interestingly and importantly, I think this effect seems to be fairly consistent across subjects. With that, I want to thank the people in my group who did the work, in particular Shabnam Kadir, Mahmoud Keshamadze, and Mikolai Kegler, as well as funding agencies. And thank you very much for your attention.
Hello, my name is Mira, and let's do some break exercise, get some energy. Alright, take a space so we can start. First, we do some shoulder walk, and at the same time you can bend on your knees a little bit. Whoop, 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 hop, 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 hop. Alright, one more time. Whoop, whoop, whoop. Alright, yes, very good. Then we do a big circle with your hands. Very big, so you can stretch your body at the same time. Up. Yes, very good. And then the another side. Up. Yes, and one more time. All right, very good. Yes. Then move your opposite knee and elbow together like this. Up. And rotate your body a little bit. Up. Your upper body. Up. 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 Yes. One more. Up. Up. Ah, very good. Then stretch your rib from side to side. Remember to breathe. All right. Good. And one more time. Yes. And the last one. You can stretch your upper body. Just relax your arms and head. And then get up and stretch your thorax. Open your. Yes. And again. Just relax. And open your upper body. Good. And one more time. Relax your body. And open your upper body. Yes, very good. We are ready. Have a nice day.
Mr. Chairman, ladies and gentlemen, I will explain you how our TPS transcranial pulse stimulation is working and which result we have achieved. So TPS is using mechanical brain tissue stimulation with shock waves. Shock waves are known in medicine since over 40 years. And the first applications on neural tissue was under the term ESWT, transcranial shockwave therapy. Now you can see here the range of intensity used for medical shockwaves treatment. It started at the very beginning for the kidney stone disintegration lithotripsy with a high intensity. Meanwhile, we are at less than 1/10 of this intensity, which is used for pain and cell regeneration, like we are using for neuroliths. Now, again, after stones unions come, soft tissue, especially pain treatment starting in 1990. And 10 years later, the tissue regeneration come to it, expanding the further use within medical use. So in the orthopedics, in cardiology, the heart stroke, aesthetics, dermatology, wound healing, urology, erectile dysfunction, and since few years now in the neurology, especially for the treatment of the Alzheimer's disease, that means of the brain. So in general, of course, it started much earlier, starting with treatment peripherally, spasticity of muscles by Dr. Lothar Busch. He also treated spinal cord injury successful, and he had a good results with unresponsive wakefulness, as you see on the attached papers. Later on, in the following years, additional indication come to it. In 2014, he started with Alzheimer's disease. And finally, in the years 2015 to 17, we performed a clinical approval study for the Alzheimer's disease treatment by TPS, as you can see on the attached paper published this year. It was together with Professor Weinsteiner in Vienna, and it resulted in CE approval of the Neurolith in 2018. Now to see what kind of pulses we are using, it's not an ultrasound, it's a shock wave. The typical parameters of a shock wave is a very steep rise of the pressure in the range of nanoseconds, slowly a falling edge, and relatively low amplitude following vibrations. And thus, on behalf of this shape, the signal contains a lot of different frequencies. And so we are talking about broad frequency spectrum. Compared to it, focused ultrasound is also a very important entity. It's using continuous train of pulses, sinusoidal pulses, as you can see on the right. And typically it started to use focused ultrasound for tissue coagulation, where you are treating, for example, cancer within the body, with contactless treatment of tissue. And on behalf of this very smooth and regular vibrations, there is only one main frequency, so the spectrum is totally different. This is basically only single frequency. So if we project now this focused ultrasound pulse on top of this shockwave pulse, you see that it looks totally different. And in addition to that, as I said, the focused ultrasound used originally for coagulation. To stimulate without heating effect, you have to interrupt the signal so that you have only bursts of sinusoidal vibrations, which are used for stimulation. Now to our application to the brain. It's a very new indication which has been prohibited in the past. And so we have made a lot of basic research on that. And we first evaluated how much pressure comes into the brain because of course, the bone is restricting the propagation of shock waves very strongly. And so we measured pressure within or after the passage through the skull. You can see here cadaver measurements in laboratory with brain tissue and also just in the water. And finally, you can see on these diagrams here, first on the right, that the shape of the focal area is relatively unchanged on behalf of the passage through the skull. But of course, the intensity, the pressure is decreasing strongly. So we have now here in the brain reduction by 65% in pressure, and correspondingly 85% reduction in energy, which is proportional to the square of the pressure. Here you see the typical TPS pulse, the shockwave pulse. We use them at maximum 25 megapascal pulses. But of course, on behalf of this attenuation effect, within the brain, we have at maximum about 9 megapascal. Now we also address the issue of hair because it's also additional source of attenuation. But fortunately, we're using enough coupling gel. We have at maximum 10%, and usually with much less hair, this is not neglectable. We had also tests to show that there are no danger at intensities used for the brain. And so we could show at animal tests with practically rats that in spite of very high intensities used, we observed no bleeding. And you can hear summarizing that we have a limitation of maximum pressure, 25 megapascal, a low energy density, and of course, the energy flux density is on maximum 0.25. Now here's a complete treatment setup. The device Neuralis, the chair where the patient is situated for the treatment. An important component of the equipment is a body track system, which allow us to document the treatment and maintain a homogeneous treatment or energy delivery. You can see here you have an infrared camera, then the reflector on the treatment part here of the spectacles for the patient, reflector on the handpiece, and this equipment helps us to show or control the treatment very precisely. We also need a calibration with this calibration tool, which is very simple at the very beginning of each treatment. Here's a typical how this treatment is performed. On the right side, you can see the information from the body track system showing how the energy is distributed and whether it is homogeneously from energy level point of view. The darker, the more energy has been applied. And on the left side, the parameters setting for the number of pulses, energy flux density, and frequency. Now to stress out the advantages of the body track system. So we are monitoring and documenting the system during the treatment real time. We have a visualization in 3 dimensions, 3 directions, 3D. And also the color is representing where the energy is applied and how much energy is being applied. And of course, we are to have it personalized and exact documentation. We are using the patient data, the MRI or CT data of his brain. Here you can see that the setup allows the treatment of the complete gray matter of the brain, of the cortex. The focal area, which is about 3 centimeters long, fits very nicely into this gray matter area or space. And so here how it looks when you are treating the patient. Of course, here the gel is missing. The areas we are treating are especially responsible for the memory functions. So it is the frontal lobe, the parietal lobe, and the precuneus. Now, the important issue is which biologic effects are introduced by using the TPS. The TPS basically heritaged all the effects proven scientifically for shock waves. So you can see here there's a process called mechanotransduction. That is basically the mechanical stimulation of biological effects. Starts with increased cell wall permeability. And here is also the blood barrier barrier opening is possible in animal at least. We have stimulation of mechanosensitive channels. And the most important biological agent, the nitrous oxides, takes care for vasodilation, increased metabolism, angiogenesis, long-term blood supply improval, anti-inflammatory effect, and further stimulation of growth factor, especially important the VGF, the ENGF, and the BDNF, which are responsible for neuron or brain tissue growth. And part of this effect are, of course, also the stimulation of stem cells. It fits very well to the known origins as far as known for the Alzheimer's disease, the deposit of proteins, inflammation, virus infection, reduced blood supply, neuroapoptosis. So it's the spectrum of TPS functions is very well fitted to Alzheimer's disease deficits. And now we performed this study mentioned at the very beginnings with Dr. Losebusch and Professor Weinsteiner, altogether 19 patients, which have been used to show the efficacy for the approval of the device. Interesting is are the first patient treated by Dr. Losebusch because we have a long-term observation possible. And so we can see in the next slide the effect of long-term effects. In parallel to that, we have a multiple placebo-controlled RCT trials ongoing. Here are the long-term results. So after the boost treatment, so the 6 sessions in 2 weeks, the green area, 3 months follow-up, and finally, in the further time, there have been maintenance treatment every 6 weeks, as shown here. And you can see that the very good improvement, TERAT score improvement of over 12% could have been more or less maintained during the first year. Here on the bottom, you see the natural decrease of brain activity when treated only conservatively. And now we have here the follow-up of second year, and unfortunately, it's visible that the improvement decreased again. And another boost-up treatment was possible to get upper again, unfortunately not at the same level at the very beginning. That shows that in the future, we have to apply this boost treatment earlier during the treatment, probably in the course of the second year. Here you can see summarized results of the clinical multicenter trial by Dr. Losebusch and Professor Weinsteiner, which is very nicely described in this paper I showed you at the very beginning. So the CERAT improvement is over 10%. You can see here the improvement of the verbal and memory functions. And finally, summarizing, we can see here that we have 6 treatments with 6,000 pulses within 2 weeks, outpatient treatment of 30 minutes only, no pain, no side effects. And we don't need any cognitive training during the treatment, only the maintenance treatments as follow-up. There is no need to shave the patient's head, and there is no need for fixation. So the TPS treatment is effective and safe. And we have also further positive outcomes for another neurological treatments in the future. Thank you very much for your attention.
Hello everyone, I hope you're doing fine despite the situation with coronavirus lockdowns, etc. I'm Koos Zevenhoven. I lead a research group at Aalto University, Finland, and I'm going to talk to you about our new brain scanner prototype that combines MRI and MEG. I will also talk about further combining it with ultrasound techniques. Let me start by bringing up this vision of a wide-ranged instrument for dealing with the human brain in clinical and basic research settings. The instrument would offer a variety of modalities for imaging as well as for stimulation and therapy. The patient or subject could sit or lie down in a fairly natural position. Here I'm going to talk about incorporating techniques related to 3 different physical principles. First, let's look at the atom. Most atomic nuclei in the body, protons in particular, have a non-zero nuclear spin. This is a quantum mechanical angular momentum which interacts with magnetic fields. It also produces a magnetic field in the surrounding space. The spin can be manipulated with applied fields and tuned to different resonant frequencies. This is called magnetic resonance or nuclear magnetic resonance. The second principle is that neuronal activity is mediated by the movement of charged ions. That means that there is an electrical current. Also, electrical current produces a magnetic field. This gives rise to MEG, the magnetoencephalogram, which is the weak magnetic field generated by brain activity. Most of this work actually relates to making use of the synergy of these 2 phenomena occurring in the brain, and to using the same sensors to detect both signals. In fact, I will also talk about influencing magnetic resonance with electric fields or currents. And if you're familiar with transcranial magnetic stimulation, TMS, or electrical stimulation, you know that also neurons respond to applied fields. But we are also adding a 3rd basic principle, which is ultrasound. That means mechanical vibrations or acoustic waves, which are above human audible frequencies. Ultrasound has been proposed for various purposes, but since this is the BrainSTIM conference, you might be interested in brain stimulation using ultrasound. In fact, ultrasound may affect both MEG signals and magnetic resonance. So there are many things here. This will almost be like a series of mini talks about different subtopics. So let's get to it. So indeed, the nuclear magnetization of a sample is based on the nuclear spin. The magnetization precesses or rotates around an applied magnetic field at a frequency that's proportional to the field. This gives rise to a signal whose phase and frequency can be controlled by additional magnetic pulses to encode spatial information. Different pulse sequences allow different types of image reconstructions. Conventionally, an MRI scanner has this fairly tight cylindrical bore. It has a persistent strong magnet, and the signal is detected by Faraday induction. One could argue that, based on these choices, MRI had sort of fallen into this potential well where the only possible direction is to go towards higher and higher magnetic fields, to 3 Tesla or 7 Tesla or even more. MRI did become the ultimate imaging modality for many purposes, but this approach to MRI also has its limitations. For example, it is costly, it is noisy, and it is incompatible with many other technologies such as MEG. So already some time ago, we finished our first proof of concept system. Our paper was well received and in fact made it to the front cover of the top journal for MRI, Magnetic Resonance in Medicine. But then there was the next big challenge. How to get from that point to something that can actually be used in practice. Even simply the ability to measure with a full sensor array from all sides of the head was going to require significant technology improvements. Unlike conventional MRI, we didn't have the backing of large corporations and there were no existing solutions to use. But we took the challenge as a research project to develop a new prototype. So many new solutions were needed. The technology differs from conventional MRI in all parts: excitation, detection, reconstruction, electronics, software, and so on. Obviously, I can't go through all the components in detail now. That will be published elsewhere. One thing to mention about the technology is that detection with superconducting quantum interference devices together with pulsed prepolarization allows using precession fields in the microtesla range. Part of the challenge is that for the extremely sensitive measurement, the total measurement noise must be very small. There are many parts that can produce noise, which easily leads to a mess. Every single component of the system must have extremely low noise. Only then you can get high quality signals. Just to bring up one of the components, here are some specs of our new pulsed polarizing coil made of type 2 superconducting multifilament wire, providing 100 millitesla of magnetic field. Close to the coil you can also see 120 sensors, which we quickly recover into their best measurable state within milliseconds from pulsing the coil. The coil itself must also be recovered by careful pulse shaping. Regarding prototype integration, we actually first worked towards an intermediate system. However, we had to move on to the final prototype before it was finished. So, finally we have a new prototype together. Publications will be coming out in the near future with more details. Let's now look at the MEG side of things. Again, magnetoencephalography, or MEG, is the measurement of weak magnetic fields generated by electrical activity in the brain. When this is done with a multi-channel measurement around the head, brain activity can be localized with the help of an electrical model based on MRI. This can, for example, be used to plan epileptic surgery. And as you can see, MRI is also useful when visualizing the analysis. So, our device uses the same sensors to measure both MEG and MRI. One thing that this allows is accurate combination of MEG and MRI data. Last year we published a calibration method for this purpose. According to the analysis, the method reaches sub-millimeter accuracy in aligning MEG and MRI coordinates. And here are some curves to show that we indeed have around 100 working sensors. On the right you can clearly see the alpha rhythm slightly below 10 Hz. Okay, so let's talk about new developments related to MR-based modeling of electrical structure in the brain. First, let's look at this study by Walters et al. About the effects of the electrical inhomogeneity and anisotropy of the brain. So for a thalamic source, we see that the resulting current pattern is severely distorted. Now this is quite an extreme case, but it suggests that inhomogeneity and anisotropy are significant and that an accurate model for measurements and stimulation should take them into account. Now what if we could map pathways of injected current within the brain to improve the models? The magnetic field due to electrical current can affect MRI phase. However, in conventional high-field MRI, the static main field drowns most of that signal. To solve this issue, we have developed ultra-low noise electronics, with which one can turn off all MRI fields within the pulse sequence. Making use of this possibility, we also developed a pulse sequence and reconstruction technique called zero-field encoded current density imaging. It's a technique that can image current pathways in volume. Using this technique, our German collaborators successfully reconstructed a volume current from phantom measurements. The next step would be to implement it in vivo. In fact, this recent study suggests that it should already be doable with the present technology. Okay, so let's move on to ultrasound. Ultrasonic fields can be focused by constructive interference of waves at the focal point. Many different uses for ultrasound have been proposed. Therapy, surgery, or temporarily opening the blood-brain barrier for drug delivery. And then there are interactions between ultrasound and magnetic resonance. But since this is the BrainSTIM conference, perhaps most interesting is neuromodulation or stimulation. The 2016 review by Rezayat et al. Cites 18 studies that successfully modulated brain activity with low-intensity focused ultrasound. 2 of these were human studies. Now, there are also some studies that dispute the notion that the observed effects are due to ultrasound directly affecting the brain. However, at least one study from last year again concluded that ultrasound was able to directly activate cortical neurons in mice. Okay, what about implementing transcranial focused ultrasound? One thing to note is the effect of the skull. As you can see in this example, the layer of bone significantly affects the ultrasonic waves, the phases and amplitudes. Constructive interference is sensitive to phase. However, this can be corrected for with the help of MRI. What we do is we first assume a homogeneous medium and then move on to correct using MRI. So this is the setup we are working on. Special care is taken to make the transducer setup non-magnetic and to prevent electromagnetic noise or interference from entering the magnetically shielded room or the squid measurement. For focusing, we use a 3D printed zone plate, in this case a Fresnel zone plate. It is basically a set of concentric rings that are positioned in such a way that the incoming plane wave from the piezo element produces a diffraction pattern that has a focus at the desired point. As soon as the coronavirus lockdowns ease a bit, we hope to be able to move forward with this. Finally, here's a little bit about cool future possibilities. It is possible to go beyond Fresnel zone plates by this holographic approach described by a Spanish group recently. For example, one could do multiple focal points, a self-bending line focus, or volumetric focusing. This can also be used to take into account inhomogeneities of the medium. To conclude, we have developed a new whole head prototype to combine MRI and MEG accurately. We have also had progress towards current density imaging of the head to improve characterization of brain activity and targeting stimulation. Finally, we are also adding focused ultrasound to the system and hope to study things like the influence of ultrasound on MEG and MR signals. So, thank you for your attention, and thank you to everyone involved.
My name is Roland Beisteiner. I'm Professor of Neurology and Psychiatry in the Department of Neurology at the University of Vienna. Since 2013, we've been working closely with Storz Medical in development activities. The new method, called TPS, transcranial pulse stimulation, has a wide range of advantages over existing techniques of brain stimulation or brain activation. What makes it special is that it opens up a whole new approach to therapy. Among the chief benefits of TPS is that it accesses deeper areas of the patient's brain than other procedures do, reaching as much as 5 centimeters into the brain. We know from functional studies that in Alzheimer's disease, it is the frontoparietal networks and the so-called default mode network that are particularly affected. The latter network plays a big part in storing and retrieving memories, things that Alzheimer's sufferers really struggle with. They find adapting to new situations very hard. Responding to changes in situations around them is extremely difficult. These are all functions performed by the frontoparietal network. That means that with this disease we already have fairly well-defined target areas we can select when activating brain tissue. We selected the frontal lobe, the parietal lobes, and the precuneus, the core network node for the default mode network. One key mechanism appears to involve stimulating the mechanosensitive ion channels. For example, this is the case for the brain growth factor BDNF and also for the vascular growth factor that leads to an increase in blood vessel growth and hence enhanced vascular supply to the brain tissue. The advantage we enjoy with this new system is the long track record of application that we have here. This technique is familiar and it is safe. One particular benefit of the existing Neuralit system is its body track function. This is a visualization and documentation system. With this software, we're actually able to precisely quantify energy inputs and precisely identify where we apply them. And all of this takes place in real time. Treatment is pain-free for patients. In a total of over 1,000 logged treatment sessions, serious side effects have not been reported once. There are 6 treatment days in total. Each treatment lasts 30 to 45 minutes, and the sessions are spread over a total period of about 2 weeks. Initial data from pilot studies reveal that highly impressive successes have been achieved with this regimen. Enhancements in neuropsychological performance. Patients frequently report that they experience subjective improvement. This may involve them noticing their memory is better. The patients become more active and less withdrawn. Social interaction is easier for them. And this also considerably improves the situation for their caregivers.
So today I'd like to talk to you about alternative methods for neuromodulation. Up till now, non-invasive brain stimulation has focused mostly on electromagnetism. So electrical and magnetic forms of stimulation really dominate the field. In our lab, we've used TMS in a variety of forms of electrical stimulation in a number of studies. We've looked at a variety of clinical disorders. We've also looked at memory enhancement, and the most technically challenging of that so far has been using closed-loop alternating current stimulation during sleep to increase memory consolidation. And we recorded brain EEG and were able to apply alternating current at the same phase and frequency as the subject's natural EEG. But the largest effect we've seen in any study so far has used TDCS that was guided by neuroimaging to produce a 2 to up to almost a 4-fold improvement in learning, a really large effect. So the stimuli we used are here. We had a series of still images. There were threats hidden in these images, sometimes snipers, bombs, things like that. People had to learn to identify them even though they were hidden. We did an imaging study and found areas that changed their level of activity as people learned to perform the task. And then we used those as targets with TDCS, and we found a doubling of improvement. We replicated this in a number of studies in our lab, and the basic design here was we obtained baseline level of expertise with still images, did training, and the training involved seeing a still image, making a yes/no response, and then the feedback was a movie. And if a threat was present and you said, no, I don't see it, then the movie showed the threat happening. This is called discovery learning. There's not much information being given, and it's a really difficult task and difficult form of learning. So we gave these stimuli to Raja Parasurman and Brian Falcone at George Mason University. They did an independent replication and found pretty much the same results that we did. Hit rate went up, false alarms came down, and D prime, a signal detection measure, more than doubled with using 2 milliamps of anodal TDCS current to the right temple versus the cathode on the left arm for 30 minutes. So this protocol and the results have been replicated a number of times. And across all these replications, we find a very consistent pattern of results. F10 is the right temple. We've also tried right parietal cortex, P4. And in all these cases, we see a significant improvement in learning relative to sham. We've also found that cathodal current over T5, this is left occipital cortex, showed improvement, but anodal current showed no significant difference. And if we compare these results to the original fMRI results, here you see the difference between threat present and threat absent after training. And areas that showed an increased response to threat present responded to anodal current, and regions that showed a decreased response responded to cathodal current, assumably reducing the level of activity in those areas. So fMRI did a great job of predicting the response to different electrode placements with really surprisingly good accuracy. So other imaging has showed us other things that might be relevant to this task. One is a series of studies by Huth et al. At Berkeley, and they found that the same area that we were targeting with TDCS responds to violent imagery. And it might have been looking for threats that produced this large enhancement. So Brian Gibson in our laboratory developed a new learning task. In this case, the task was to look at a series of images like this, and you had to identify what country each picture was taken from. Again, a very difficult task, not a lot of information given to make it work. But using the same protocol, we were able to get almost a 4-fold improvement in learning. Again, a really big effect. So from baseline through individual training blocks and then the post-test, people were able to improve both with anodal current and with cathodal current. So less of an improvement with cathodal current, but it was still there relative to sham. We've done a number of follow-on studies using this F10 TDCS protocol. And we're learning more about it, what it does, why it works, and so on. But there are other things besides learning that we want to tackle with neuromodulation, and one of them is addiction. So we had a paper come out in 2014 where we had tested 45 patients recovering from stimulant dependence. We tested them at about 1 month of abstinence and then followed them for up to 6 months. And at that time, after 6 months, about half of this population had relapsed to drug use. The other half had maintained abstinence. We compared the bold response that we got at 1 month of abstinence to see if there's anything in their brain function that predicted relapse. And it turned out that there was. There's an area in posterior cingulate cortex that predicted relapse with close to 80% accuracy. And adding a history of mania improved this accuracy to over 90%. So, and 84% using cross-validation. So it's a fairly good way, a good predictor of relapse. That suggests that activity in this part of the brain might be important for relapse behavior and addictive behavior in general. And if you look in the literature, most psychiatric illness includes cingulate or limbic system activity that has something to do or appears to have something to do with different components of those illnesses. So how do we target deep CNS areas without disturbing more superficial areas? There are methods for using deep TMS, but really they stimulate the whole brain simultaneously. So what are the basic premises of neuromodulation? It's that behavior depends on the nervous system function. The nervous system is a physical system, therefore functional physical changes in the nervous system should be able to change behavior. So how do we produce those changes? Before you try something, it's good to assess all your tools. You might just have a few, but it could turn out you have many more than you knew. So for neuromodulation, what are our tools? Really, our tools are energy. Energy is transmitted as forces, and there are 4 fundamental forces that we know about so far. The weak and strong force that have very short ranges and mostly have to do with holding molecules and atoms together. There's gravitation that acts across great distances, but is relatively weak, and electromagnetism. So because the weak and strong force act across such short distances, they're really not useful to us. And gravity, being relatively weak, and we don't have a good way to control it really, also isn't useful. So really, we've been doing the right thing. Most of useful forces are electromagnetic. If you look at energies derived from forces, there are a number of them. Some only act at very small or very large distances, so they're not really useful to us. So macroscopic energies, this remaining 10, are energies that we might be able to use to alter brain function. Chemical energy, the energy in chemical bonds, basically chemistry, are what pharmaceuticals depend on, and we've been using those for years. But we can't really direct them to specific brain areas without influencing most of the rest of the body. Electromagnetism, again, we've used that thus far, electric ionization and magnetic energies. But we're looking for something new, a better way to do this, or a different way to do this. So the remaining energies that we have are elastic, mechanical wave and sound waves, which together explain ultrasound and its effects, elastic and mechanical energy, which is simple physical pressure, radiant energy, which is light, and thermal energy, which is heat. Now, most of these energies are actually acting through electromagnetism. And using these energies, we can influence electromagnetism indirectly by through these different forms of energy. So the first one I want to talk about today are mechanical waves and sound. So using ultrasound. These are kinetic and potential energies moving through different kinds of media. It's actually been known for almost a century that high frequency sound, what we now call ultrasound, can affect biological processes. So Newton Harvey found this out and published in 1929. About 30 years later, William and Frank Fry, a pair of brothers doing biological experiments, showed effects in the CNS directly using ultrasound. And here's just a little figure from that science paper that they published in 1958, showing that there was a reduction in visual evoked potentials using ultrasound. So, and Harvey's original work was in using frogs as an example. And by applying ultrasound to frogs, he was able to modify heart rate and other physiological processes. So this is a relatively old method, it turns out. Why should we be interested in ultrasound? First of all, is anatomic specificity. So with regular TDCS, not HT-TDCS, but regular TDCS, there's relatively broad swaths of tissue that are stimulated or altered with electric current. And with TMS, again, a relatively large area. With ultrasound, it turns out that modeling suggests you can get less than a centimeter of stimulation. So a relatively discrete and precise area of stimulation if you set up the experiment in the correct way. TDCS can have deep effects, but it's impossible to control precisely because electric current follows electromagnetic laws as it moves through the brain. And it's really difficult to force it to move one way or another using just surface electrodes. Ultrasound, on the other hand, by using multiple emitters all focused on the same area, can produce relatively weak effects until those different waveforms converge in a specific location. So our first study used a diagnostic imaging system and used a diagnostic imaging protocol with ultrasound, the same kind of protocol that we use in to measure and monitor fetuses in the womb. Doing this, Ben Gibson and Jay Sanguinetti in my lab applied 2 minutes of this imaging ultrasound protocol to the head and monitored brain excitability using TMS-evoked MEPs. And what they found was about a 30% improvement in MEP amplitude, or a 30% increase after 2 minutes of TES that lasted about between 6 and 11 minutes after stimulation. So we found, and other groups have found, that ultrasound can, applied to the brain, can affect brain function. How might we use this to impact drug use disorders? Well, we've started a series of studies where we used a focal ultrasound emitter positioned over parietal cortex, but directed towards the posterior cingulate cortex, where we found that big predictive effect of relapse. And in a series of pilot subjects, we identified the target location relative to the scalp location, positioned the emitter for each individual to direct the ultrasound towards posterior cingulate. And then you can see the ultrasound setup here. There's a system that generates the ultrasonic waveform in a controlled way. It's amplified, and then an oscilloscope can be used to monitor the ultrasound, and that's sent to the emitter. And this shows a bit of how the oscilloscope sees the ultrasound coming out. So basically, every few seconds, there's a tiny pulse of ultrasound over the course of about 10 minutes. The reason to space this out is partly heating, that we want to minimize the amount of heating being produced. So giving spacing between ultrasound pulses minimizes the amount of heat. We'll see later this might actually be part of the mechanism of the effect. So it might be something to think about doing it more quickly, but we're trying also to stay as far away from the safety limits as we can get. So we measured the effect on the brain using spectroscopy. And spectroscopy is an MRI-based method that can look at the concentrations of different metabolites in the brain. And by doing this, you can see for posterior cingulate, our target, but also other brain areas, what the effects are. And there's a method called EPSI that produces 3D MR spectroscopy. So it allows us to simultaneously see across the whole brain. And this allows us to look and see how large the effects are from ultrasound. And this just shows on a Siemens how we set up this protocol, this EPSI protocol. What we found were reductions in creatine, choline, and a combination of glutamate and glutamine, all within a centimeter or two of our target. So creatine is related to metabolism, also GLX, this combination of glutamate and glutamine, related to neurotransmission, and choline is related to membranes. All of them reduced a little bit during stimulation. So in conclusion, we found that the effects of ultrasound, some of them have been known since the late 1920s. It's thought to be safe if used below certain thresholds. Neuromodulation applied in those thresholds produces changes in the brain that we can see. So now we're planning to use this in a series of studies, first to see if we can reduce smoking. But given that nearly all forms of psychiatric illness involve deep brain structures, this may prove to be a very effective method for altering brain function in a way that will produce improvements in behavior in different psychiatric illnesses. How else can we use energy to alter brain function? Radiant energy is another way. So using light. And light therapy has actually been practiced for thousands of years. It's not clear if the early forms of it worked. Light therapy was developed for biomodulation in the Soviet Union, now Russia, as early as 1964. And there's a nice summary article from 2017 that you can look up if you're interested to learn more about this. Today, there's a number of commercial methods available. One of them is made by a company called V-Light, and they make a device called the Gamma Neuro. So the Gamma Neuro uses 810 nanometer pulses. It's pulsed at 40 hertz for about 20 minutes. And it uses LED emitters that are spread around the scalp, and one goes up the nose. And we use that same threat target detection task that I described to you earlier to see what the effects for the V-Light might be. So we use the same task, again, a baseline training and test. And during the training, we either applied infrared light in that 40 hertz pulse manner or a sham. And we looked at the change in performance with training. We found that using this, we again got about a doubling of learning, applying infrared light for 20 minutes with a reasonably large effect size and Cohen's d of 1.4. So a fairly large effect. We wanted to replicate this, and we got a new Gamma Neuro device that had slightly different properties and tried to replicate and saw no effect. We went back to the original device and found that, again, we got a reasonably large performance improvement. So what was going on? It turned out the difference between these two devices was that for the original device, the posterior and anterior phase of light was 180 degrees out of phase. So the back system would pulse, then the anterior LEDs would pulse, and back and forth. The second system, both lights pulse simultaneously. So we asked them to make a new device that pulsed out of phase again. Tried that again, but lo and behold, no effect. So it may be that that initial result was spurious and just a statistical anomaly. But we're trying a third time using those original devices again, but this time in a double-blind manner. So two possibilities are, there's something about that original device that produced this relatively large change in behavior, or it's the fact that when we ran this first, we used a single blind method. So the experimenter knew the condition. In the second and third attempts, the experimenter didn't know the condition. So we're going back to those original devices to see if we can get the same effect. So this is still work in progress. I don't know what the end result's going to be. Hopefully when the coronavirus lockdown ends, we'll be able to get back into the lab and try this. What's another form of energy that we might use? Another one is mechanical energy, physical pressure. So this is a little different than these other forms of energy in that you can't really apply pressure directly to the brain safely, but you can apply it to the body, basically to the peripheral nervous system. And over the years, there have been a few different methods that have been developed to be able to use pressure in a way to alter behavior. For a long time, we've known that humans like to bite when either they're trying to produce strength, when you're doing lifting or something like that, you tend to bite down, but also for pain. And for years, people would bite when they were in pain. So when they did surgery, before they had anesthetics, they would give you something to bite on, or when women were in childbirth, they would give them something to bite on. Other places in Asia, there were methods of acupressure developed where just pressing on the body was thought to produce changes in how body function and behavior. Another method is one by Temple Grandin developed. Temple is a woman with autism, but very smart. And she realized working with cows that they had a machine to hold cows while they did work on the cow, and the cow would calm down. She tried it on herself and found that she calmed down. So there are a number of methods in the past that have used pressure. However, in the modern day, there's a new method that is in development. And this uses pressure on the teeth and jaw to treat neurological disorders. Brendan Stack and Anthony Sims are two there's actually a number of dentists now working on this, but Brendan Stack was the first. And what he saw was that when he treated teeth and jaw disorders in patients that also had neurological disorders, a number of times that neurological disorder would improve. And follow-up work by Anthony Sims and other dentists have shown this to be true, that treating dental problems can actually have an impact on the nervous system. Now, I learned about this because of a neurological disorder that my son Ryan developed a number of years ago. So he was a great, wonderful kid. And as he was growing up, started to do a lot of physical things. One day in gym class, he twisted his knee and his ankle and knee. And he had to use a pair of, well, he couldn't walk very well. So he was either in a wheelchair or used crutches for a while. But he got better. And then a few months later, developed a pain syndrome. And this syndrome involved a lack of blood supply to the affected leg. And you can see the difference in color here. It caused a lot of pain for him. We took him to a doctor. The doctor gave him medications that helped with the pain, but it produced a really horrible side effect where he would develop tics. He would just fall down and go into seizures. Sometimes he would get dystonia where he had real trouble moving. And eventually we were able to treat all this. And as of today, he's doing great. He was valedictorian for his high school class. He's in an honors engineering undergraduate program at Arizona State. We got through this, but how did we do that? It turned out that by adopting the methods developed by Brendan Stack and other dentists, we were able to get him to the point where we could calm down his tics and reduce his pain. So we came up with a non-pharmacological method to treat his pain and his motor symptoms. And this shows by just having him bite down on something, he was able to get up and walk for the first time in over a month, just using a stack of bite guards that he put in his mouth. So again, it was difficult, but not impossible for him to walk. And other patients have
My
shown
symptoms were twitching, I had barking noises, I'd twitch my neck back and forth, cause myself whiplash. I mean, I've always looked at people and just wishing that I could just stay still.
So that was a patient that had Tourette syndrome, and just by biting on a orthotic, a mouth guard, he was able to reduce his symptoms. And other patients with a variety of symptoms. Here, this patient has blepharospasm, here Tourette syndrome, here dystonia, and here tics and dystonia after CRPS, like my son Ryan. All of them benefited by just putting a stack of tongue depressors in their mouth. How does this work? There are three likely scenarios. One of them is it's purely psychological, that there's no neurological basis to this, that for whatever reason, people are able to have their symptoms go away just by the belief that they would be helped. Another is that there's something special about the temporomandibular joint in that when it's irritated or pain is caused by a problem with the jaw or teeth, that this somehow propagates into the nervous system and causes effects that part of those are changes in brain networks, and that by having them bite, this normalizes those brain networks and reduces pathology. A third possibility is that the nervous system, the peripheral nervous system in the jaw, is actually designed in such a way that it's sensitive to pressure on the jaw and teeth and is designed to alter nervous system behavior that way. So we've done a number of studies, one of them looking at MRI. And while people have one of these devices in their mouth, you see that the condyle, the end of the jaw, moves out of the articular eminence and away from, assumably, where the nerves that innervate the jaw tend to run. So it's taking pressure off of those nerves. We've also done fMRI, and we found that using devices of different thicknesses and have people doing a motor task while they bite down on these devices, the thicker device, the larger the effects in the brain of biting down. So this uses a very thin device as a sham device and a thicker device as an effective device. And you see more effects in the brain as people bite down on thicker devices. And the largest effects of these are in the cerebellum. We've also done studies with blinking and find that, again, with clenching an effective device during blinking versus rest, that you see effects in the brain when they're clenching versus not clenching. And the interaction of these, so movement, blinking versus not blinking, and clenching versus not clenching, looking at that interaction, there's a significant effect across a variety of brain areas, focused in the caudate, a little bit in the cerebellum, but also a bit in frontal and parietal cortex as well. So we're finding effects here in the brain using these methods. We published a short paper in Pain Medicine on this method. And as of today, there's many remaining questions about it. If tics are in all muscle groups, why should biting, that is dealing specifically with the jaw, have any effect on these other muscle groups? Why are we wired that way if it's true? How would problems with the trigeminal nerve, say from temporomandibular joint illness, lead to changes in brain function that's seen with fMRI? And assuming that overstimulation is causing these problems, why does it selectively cause just some motor illnesses and not others? How are genetics involved? And these illnesses tend to wax and wane over a patient's lifetime. Why is that? Are these changes in the jaw that are causing these problems? So there's a lot of questions. So in conclusion, there's a number of older technologies to affect neurological symptoms and behavior. Oral orthotics are a relatively new technology, but along the same lines. Dentists working with patients have found that patients with Tourette's, Parkinson's, dystonias, and complex regional pain syndrome, like my son Ryan had in the past, all can benefit from oral orthotics, oral appliances. So, and we found some consistent fMRI effects so far, suggesting that there are actually brain effects of using these. So if we understand this better, this might provide many new and more effective treatments for dystonia, chronic pain, and other forms of illness. Finally, and really quickly, heat may be an important property of, or an important side product of a number of forms of neuromodulation that alter brain function. And there's a number of papers coming out showing that different forms of stimulation, like kilohertz frequency, brain stimulation, and ultrasound, might both have heat as a mechanism of effects on brain function. So we haven't, in my lab, done any studies like this yet, but there could be a number of effects that we need to look at that involve heat. So what's the future of non-invasive brain stimulation? Really, it's a combination of developing new technologies and making older technologies more effective. But I believe that the future of non-invasive brain stimulation is really us looking at every energy possible, looking at its advantages and disadvantages, and seeing which of these might ultimately produce positive effects in the brain. And the future, besides finding new methods, I continue to believe involves neuroimaging. So knowing the physical basis of a cognitive process and how it goes awry in different illnesses will guide us in the application of non-invasive brain stimulation. And the more that we can incorporate neuroimaging into this, the better we will do. So having more tools in our toolbox allows us a greater variety of methods to correct problems indicated by neuroimaging. We still have a lot of work to do, but I'm hoping everyone watching this that is involved in this research will understand that the more we can incorporate into our toolbox, the more we'll be able to accomplish. So finally, thanks to many, many people that have been involved in these studies and our granting agencies that have been very helpful in performing these studies. And thank you for listening.
Ei, miksi se jääty? Se on jääty se kuva.
Okay, hello everyone. It's the BrainSTIM 2020 studio again here. I'm Jaakko Nieminen.
I'm Aino Tervo. Hello.
And this is now the closing ceremony of our meeting. Welcome to this session. And in this session, we are going to present the Young Investigator Awards that we are going to share and to the best oral presentations and to the best poster presentations.
So first, the best oral presentation that will be given to the presentation with high scientific quality that is presented in an excellent way. And the best oral presentation is by Daniel Keser, worth 500 euros. Congratulations, Daniel.
Very good. And then we had the other category, that was the best poster presentation. And this was tough questions as there were many excellent posters, and the jury decided that the best poster presentation that's going to be shared. So there will be two certificates, and those go to Heikki Sinitalo and Karole Vagnon. Congratulations, Heikki and Karol.
Yes, so congratulations also for the best poster presenters from my side. And now, in the end, from the studio, on behalf of the organizers, we want to thank everybody, the conference chairs, Vincent Clark, Risto Ilmoniemi, all the presenters, all the attendees, and all the team members of the local organizers. So thank you, everybody. We hope to get some feedback from you, so we will soon post a link to our feedback form in Twitter. So please send us feedback. What did we do well, and what can we improve?
Very good. And now it's then time to move on and listen to the final words by Vince Clark. Let's start Vince's presentation.
I hope you had a great BrainSTIM meeting. We hope to do this again next year, and we hope to see you in person. Please let us know what you thought about this year. If you had anything you liked or didn't like, please get in touch, and we'll see you next year. Bye.